So, hello students, welcome to Karnataka. This is Baswaraj, your biology master teacher. I hope everyone is safe and sound and doing extremely well, students. Now, this is the one-shot lecture on principles of inheritance and variation. Each class will now all the way from the starting part, that is monohybrid cross, dihybrid cross we'll be doing till entire diseases in one shot in one lecture. Okay, that is principles of inheritance. Now, students, each class na start madakina munche, I want everyone to run and get three things. That is, first thing is get a pencil. Get one pencil to write things. So if you want to write, you need a sheet of paper or a book. So one book tomani, one pencil tomani. Adadmele, I want everyone to carry one water bottle also. So it will be easy for you to rehydrate yourself during the class. Nidda kuda barala. Sarina. So that is the starting of the class, students. I want everyone to go quickly run and get the items. That is one short lecture on principles of inheritance and variation. Now, the first thing which I want to tell you is the weightage. Chapter important. For example, students, this chapter right here, principles of inheritance and variation. Every single year, KCET exam, every single year, you will get around 3 to 4 questions. Every single year, 3 to 4 questions, pakka, guarantee. 3 to 4 questions. Sometimes it can also increase to 5 also, depending upon the mood of the paper setter. So generally 3 to 4 questions each chapter in the baratte. And also students, board exam students goskara kuda, if you are from board exam, PUC board exam idre, very high weightage chapter. That from this chapter itself you will get high weightage questions. So watch the video till the very end, understand every single word I'm teaching you. If you have any doubt anywhere, you can always let me know in the comment section and I will reply to your comments. And so with that being said, students, let's start the class. Okay. Now students, ನೀವು ಯಾವತ್ತಾದ್ರೂ ಕೂಡ ಒಂದು ಫ್ಯಾಮಿಲಿ ಫಂಕ್ಷನ್ ಹೋಗಿರ್ತೀರಾ ಅನ್ಕೊಳ್ಳಿ. ಆ ಫ್ಯಾಮಿಲಿ ಫಂಕ್ಷನ್ ಹೋಗ್ಬೇಕಾದ್ರೆ ಒಂದು ರಿಲೇಟಿವ್ಸ್ ನಿಮ್ಗೆ ಸಿಕ್ಬಿಡ್ತಾರೆ. ಆ ರಿಲೇಟಿವ್ಸ್ ನಿಮ್ಗೆ ಕೇಳ್ತಾರೆ, ಬೈಲಮ್ಮ ಬಾಯ್ಲಿ, ನಿಮ್ಮ ಮೂಗು ನಿಮ್ಮ ಅಪ್ಪ ತರ ಇದೆ, ನಿಮ್ಮ ಕಣ್ಣು ನಿಮ್ಮ ಮಮ್ಮಿ ತರ ಇದೆ, ನಿಮ್ಮ ಹೈಟು ನಿಮ್ಮ ಅಪ್ಪ ತರ ಇದೆ, ನೀನ್ ನಡೆಯೋ ರೀತಿ ನಿಮ್ಮ ಮಮ್ಮಿ ತರ ಇದೆ. ಆತರ ಹೇಳ್ತಾರ ಯಾರಾದ್ರು? Do they always tell you that your face looks like your father? You are almost looking like your father. You almost resemble your father or mother. ಹೇಳ್ತಾನೆ, ಕೇಳಿರ್ತಾನೆ. That is called as inheritance, student. That is called as your inheritance. Now understand students. Understand all of you. Have you ever noticed all of us look and sound like our parents or grandparents? Now you can ಹೇಳದ್ನಲ್ಲ, ನೋಟಿಸ್ ಮಾಡಿರ್ತೀರಾ. That is from your parent to the offspring. Parent ಇಂದ ನಮ್ಗೆ certain information transfer ಆಗ್ತಾ ಇದೆ, and because of that information both your parents and you almost look alike. ಏನಾಗಿ behavior ಕೂಡ ಆಗಿರಬಹುದು, which you pick up later on. Okay, so that is called as your inheritance. That is a phenomena by which physical, physiological and behavioral features of parents are passed down to offsprings generation after generation is called as inheritance. It is called as inheritance. ಆಮೇಲೆ ಯೋಚನೆ ನೋಡಿರ್ಬೇಕು, ನೇಚರ್ ಕೂಡ always a cat will give birth to a kitten, a dog will give birth to a puppy, a elephant will give birth to a elephant calf. But ಯಾವತ್ತೂ ಕೂಡ ನೇಚರ್ ಅಲ್ಲಿ ಒಂದು ಕ್ಯಾಟ್ ಒಂದು ಜಿರಾಫ್ ಬರ್ತ್ ಕೊಡಕ್ ಆಗಲ್ಲ, ಆತರ ಸಾಧ್ಯನೇ ಇಲ್ಲ. ಆ ಕ್ಯಾಟ್ ವಿಲ್ ಆಲ್ವೇಸ್ ಗಿವ್ ಬರ್ತ್ ಟು ಅ ಕಿಟನ್ ಕೂಡ. ಸರಿನಾ? ಜಿರಾಫ್ ಇಂದ ಒಂದು ಲೈನ್ ಬರಲ್ಲ ಯಾವತ್ತು. ಓಕೆ, ಸೊ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಇಸ್ ಬೇಸಿಕಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಆಫ್ ಇನ್ಫಾರ್ಮೇಶನ್ ಫ್ರಮ್ ದ ಪೇರೆಂಟ್ ಟು ದ ಆಫ್ ಸ್ಪ್ರಿಂಗ್. ಹ್ಯೂಮನ್ಸ್ ಇಂದ ಹ್ಯೂಮನ್ಸ್ ಗೆ ಮಾತ್ರ ಆಗುತ್ತೆ ಅದು ಕೂಡ. ಇಟ್ಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಲೈಕ್ ಬಿಗೆಟ್ಸ್ ಲೈಕ್. ಓಕೆ, ಲೈಕ್ ಬಿಗೆಟ್ಸ್ ಲೈಕ್. ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್, ಓಕೆ, ನೀವು ನೋಟಿಸ್ ಮಾಡಿರ್ಬೋದು, ಓಕೆ, ನಿಮ್ದು ಕಣ್ಣು ಮೂಗು ನಿಮ್ಮ ಅಪ್ಪ ತರ ಇದೆ, ಬಟ್ ಅದೇ ಸೇಮ್ ಟೈಮ್ ನೀವು ಎಕ್ಸಾಕ್ಟ್ ಕಾಪಿನ ಅವ್ರದ್ದು ಇಲ್ಲ. ಆ ಸ್ವಲ್ಪ ಚೇಂಜಸ್ ಇರುತ್ತಲ್ಲ, that small changes is called as variation. So transferring of information is called as inheritance. A small change in the copying time alli, a small changes is called as variation. And that is your chapter name, inheritance and variation. ಈ inheritance ಹೆಂಗಾಗುತ್ತೆ, ಈ variations ಹೆಂಗಾಗುತ್ತೆ, that is what we will be studying in this chapter. That is principles of inheritance and variation. Is a degree by which progeny differs from their parent. Parents in the offspring, right? Sorry, offsprings parents in the ಎಷ್ಟು ಡಿಫರೆಂಟ್ ಇದ್ದಾರೆ, ನಮ್ಮ ಅಪ್ಪ ಇಂದ ನಾನು ಎಷ್ಟು ಡಿಫರೆಂಟ್ ಇದೀನಿ, that is called as variation. We'll be studying this chapter. Okay, so inheritance and variation. Inheritance is the process by which characters are passed on from the parent to the progeny based of heredity. Each each child inherits a unique set of genes from their parents. ನಿಮ್ಗೆ ಯಾರು ಗೊತ್ತು? For example, if I'm XX, one X chromosome from mother, one X chromosome from father. If I'm XY, one X chromosome from mother, Y chromosome from father. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ದಟ್ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಇದೆಯಲ್ಲ, ಆ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಏನೋ ಇನ್ಫಾರ್ಮೇಶನ್ ಇದೆ, ಆ ಇನ್ಫಾರ್ಮೇಶನ್ ಇಸ್ ಬಿಂಗ್ ಟ್ರಾನ್ಸ್ಫರ್ಡ್ ಫ್ರಮ್ ಪೇರೆಂಟ್ ಟು ದ ಆಫ್ಸ್ಪ್ರಿಂಗ್. ಸರಿನಾ? ದಟ್ ಈಸ್ ಯುವರ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂಡ್ ವೇರಿಯೇಷನ್. ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ಇನ್ಹೆರಿಟೆನ್ಸ್ ವೇರಿಯೇಷನ್ ಯಾವಾಗ ಆಗುತ್ತೆ ಅಲ್ಲ? ಓಕೆ, ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ದೇರ್ ಆರ್ ಟೂ ಟೈಪ್ಸ್ ಆಫ್ gene chromosomes in your body. One is called as autosomes, one is called as sex chromosomes. ಯಾವತ್ತೂ ಕೂಡ ಈ ಸೆಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತಲ್ಲ, ಆ ಸೆಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಆರ್ ಫಾರ್ಮ್ಡ್ XX, XY, ದೇ ಆರ್ ಫಾರ್ಮ್ಡ್ ಆಸ್ ಅ ರಿಸಲ್ಟ್ ಆಫ್ ಮಿಯೋಸಿಸ್. ಆಟೋಸೋಮ್ಸ್ ನಾರ್ಮಲ್ ಮೈಟೋಸಿಸ್. ಸೆಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಈ XX ಮತ್ತು XY ಇದೆಯಲ್ಲ, ಸ್ಪರ್ಮ್ ಮತ್ತು ಎಗ್, ಈ ಎಲ್ಲಾ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತಲ್ಲ, ಈ ಫಾರ್ಮೇಷನ್ ಆಗೋದು ಬೇಸಿಕಲಿ ಆನ್ ದ ಬೇಸಿಸ್ ಆಫ್ ಸೆಕ್ಸ್ ಬಿಕಾಸ್ ಆಫ್ ಮಿಯೋಸಿಸ್. ಸರಿನಾ? ಸೊ ಮಿಯೋಸಿಸ್ ಇಂದ ಆಗುತ್ತೆ ಇದು. ಸರಿನಾ? ಸೊ ಮಿಯೋಸಿಸ್ ಆಗ್ಬೇಕಾದ್ರೆ ಏನಾಗುತ್ತೆ, ದೇರ್ ಇಸ್ ಕ್ರಾಸಿಂಗ್ ಓವರ್, ಅದಕ್ಕೆ ವೇರಿಯೇಷನ್ ಬರುತ್ತೆ. ಅವತ್ತು ಇಲ್ಲಿ ವೇರಿಯೇಷನ್ಸ್ ಇರುತ್ತೆ. ಸೊ ಸ್ಪರ್ಮ್ ಪ್ಲಸ್ ಎಗ್ ವಿಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಝೈಗೋಟ್. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತಿದ್ದು ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುತ್ತೆ. ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್ ಈ ಎಲ್ಲ ಸೈನ್ಸ್ ಇದೆಯಲ್ಲ, ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆಗಲಿ ವೇರಿಯೇಷನ್ ಆಗಲಿ, ಆರ್ ವಿ ಯೂಸಿಂಗ್ ದಟ್? ಎಸ್, ವಿ ಆರ್ ಯೂಸಿಂಗ್ ಇಟ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಈ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಮತ್ತು ವೇರಿಯೇಷನ್ ಇದೆಯಲ್ಲ, ಇಟ್ ಈಸ್ ಮೇನ್ಲಿ ಅಬ್ಸರ್ವ್ಡ್ ಇನ್ ಸೆಕ್ಷುಯಲಿ ರೀಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಆರ್ಗಾನಿಸಮ್ಸ್. ಈ asexual reproduction, they will produce clones. But this inheritance and variation, it is mainly observed in sexually reproducing organisms like humans, like cow. So if I know ಒಂದು ಕೌ ತುಂಬಾ ಚೆನ್ನಾಗಿ ಮಿಲ್ಕ್ ಕೊಡುತ್ತೆ, ಬಟ್ ಆ ಕೌ ತುಂಬಾ ಬೇಗ ಡಿಸೀಸ್ ಬಂದ್ಬಿಡುತ್ತೆ ಕೌಗೆ. I have one more cow, ಮಿಲ್ಕೇ ಕೊಡಲ್ಲ, ಬಟ್ ಇಟ್ ಇಸ್ ವೆರಿ ಹೈಲಿ ರೆಸಿಸ್ಟೆಂಟ್. What will I do now? I will take this cow, ಎರಡು ಮಧ್ಯೆ ಒಂದು ಬೇಬಿ ಬರ್ಸ್ತೀನಿ, ಆ ಬೇಬಿ ವಿಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಅ ಲಾಟ್ ಆಫ್ ಮಿಲ್ಕ್ eventually and it will also be disease resistant. That is called as artificial selection. One example ಕೊಡ್ತೀನಿ, ಎಕ್ಸಾಂಪಲ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. I have one mango plant which is producing a lot of mangoes, but mango sweet ಇಲ್ಲ. Okay, but I have one more mango tree, it produces very few mangoes but ತುಂಬಾ ಸ್ವೀಟ್ ಆಗಿ ಕೊಡುತ್ತೆ mango. So what do? Select the particular this plant, ಎರಡು ಕಂಬೈನ್ ಮಾಡಿ, we get a new plant. A new plant ಅಲ್ಲಿ lot of mangoes, all the mangoes are sweet also. That is called as artificial selection or artificial hybridization. ಈ ಎಲ್ಲ ಮಾಡೋದು ಕೂಡ ನಾವು humans. Okay, one example ನಿಮಗೆ NCERT ಅಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ ಇಲ್ಲಿ. For example, through artificial selection and domestication of ancestral wild cows, we have well known Indian breed, that is Sahiwal cows in Punjab. Right, Sahiwal cows in Punjab is a product of your variation and inheritance. Okay, ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಈಗ ಈ ಎಲ್ಲ ಸೈನ್ಸ್ ಇದೆಯಲ್ಲ, ಇದು ಮುಂಚೆಯಿಂದ ಕಾಲದಿಂದ ಬರ್ತಾ ಇದೆ. ಫ್ರಮ್ ಮೆನಿ ಇಯರ್ಸ್ ವಿ ಹ್ಯಾವ್ ಬಿನ್ ಪ್ರಾಕ್ಟಿಸಿಂಗ್, ವಿ ಹ್ಯಾವ್ ನೋನ್ ಅಬೌಟ್ ಇನ್ಹೆರಿಟೆನ್ಸ್, ವಿ ಹ್ಯಾವ್ ನೋನ್ ಅಬೌಟ್ ವೇರಿಯೇಷನ್. ಬಟ್ ಈ ಆಕ್ಚುಯಲ್ ಸೈನ್ಸ್ ನ ಓದಿದ್ಯಾರು? ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ಆ ಸೈಂಟಿಸ್ಟ್ ಹೆಸರೇ ಗ್ರೆಗರ್ ಮೆಂಡೆಲ್. ಸರಿನಾ? ಸೊ ಥ್ರೂ our ancestors knew about the inheritance and characters of variation. It was during the 19th century we worked on and scientific basis of this phenomena was finally found out. And it was Gregor Mendel conducted hybridization experiment. Barada idike. What is hybridization? Hybridization experiments are basically monohybrid cross, dihybrid cross, all of them. Okay, on garden pea plants mele for 7 years, very important line, for 7 years, from 1856 to 1863, 7 long years he studied about your pea plants or hybridization in pea plants for 7 long years. Okay, Gregor Mendel. Okay, and proposed the law of inheritance in living organisms. So he gave us something called as law of inheritance. And law of inheritance, there are 3 main laws. The first one is called as law of dominance, second one is called law of segregation, and the third one is called as law of independent assortment. ಈ 3 ಲಾನು ಕೂಡ ನಾವು ಇವತ್ತಿನ ಕ್ಲಾಸಲ್ಲಿ ಕಲಿಯೋದ. Sarna, that is called as Mendelian genetics. This entire thing is called as Mendelian genetics. Sarna, during Mendel's investigations into inheritance pattern, it was for the first time that statistics analysis, mathematics was used. Usually, ಬಯೋದಲ್ಲಿ ಮ್ಯಾಥ್ಸ್ ತುಂಬಾ ಕಮ್ಮಿ ಯೂಸ್ ಮಾಡೋದು, ಈಗಲೂ ಕೂಡ ಮುಂಚೆನೂ ಕೂಡ. But Gregor Mendel was one of the first scientists who introduced mathematics and statistics into ಬಯಾಲಜಿ ಕೂಡ ಸರಿನಾ. ಸೊ ಯಾರಾದ್ರೂ ಬೈಬೇಕಂದ್ರೆ ಇವ್ರಿಗೆ ಬೈ ನೀವು ಸರಿನಾ. ಮ್ಯಾಥ್ಸ್ ಕೂಡ ಬಯಾಲಜಿಲೂ ಮ್ಯಾಥ್ಸ್ ಬಂದ್ಬಿಡ್ತು ಈಗ ಸರಿನಾ. ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್ now here are few things which you need to know about his experiment. His experiment had a large sample size. ನೋಡಿ ಯಾವತ್ತೂ ಕೂಡ ಏನೇ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಬೇಕಾದ್ರೆ ಇಫ್ ಯು ವಾಂಟ್ ಮಿ ಟು ಬಿಲೀವ್ ದಟ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಇಸ್ ಗುಡ್ ಇಟ್ ಕ್ಯಾನ್ ಟ್ರಸ್ಟ್ ವರ್ದಿ, ತುಂಬಾ ಸ್ಯಾಂಪಲ್ ಸೈಜ್ ಮಾಡಬೇಕು. ಅಂದ್ರೆ ಯು ಹ್ಯಾವ್ ಟು ಮೇಕ್ ಮಲ್ಟಿಪಲ್ ಕಾಪೀಸ್ ಆಫ್ ಇಟ್ ಅಂಡ್ ಮೇಕ್ ದಿ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಇನ್ ಅ ವೆರಿ ಲಾರ್ಜ್ ಸ್ಕೇಲ್. ಅವಾಗಲೇ ಮಾತ್ರ ಯುವರ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಹ್ಯಾಸ್ ಅ ವ್ಯಾಲ್ಯೂ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ನಾವು ಒಂದು ಚಿಕ್ಕ ಪ್ಲಾಂಟ್ ಎಕ್ಸಾಂಪಲ್ ಮಾಡ್ತೀನಿ, ಒಂದೆರಡು ಪ್ಲಾಂಟ್ ಮಾಡ್ತೀನಿ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. somehow I get good result, but I can't take that for final result because I need a large sample size and I need to replicate that work multiple times. Then only my work is final and good work. Now also confirmation of his inferences from experiments on successive generation after generation he did the work. Mendel investigated characters in a garden pea plant that were manifested as two opposing traits. Now students, what's a word? This trait. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. There is something called as a character, there is something called as a trait. What is the difference between a character and trait? Character is the individual phenotype. For example, height, weight, handwriting. But am I tall? Am I short? Am I fat? Am I thin? My handwriting is very good, handwriting is very bad. That becomes a trait. So height, weight, handwriting. Or one more example ಕೊಡ್ತೀನಿ. Color of the flower, that is a character. But now I'm not telling which kya color anta helilla nanu. When I mention the type of color, that becomes a trait. So color of a flower height, that becomes a character. Red color, purple color, pink color, that becomes a trait. So trait is always in multiples. Trait is a multiple phenotypic expression of a character. Okay. Now, this allowed him to set up a basic framework of rules governing the entire instruction. Okay. Now, Mendel performed hybridization. Understand, students, what is the meaning of hybridization? Students, there are three main steps in hybridization. Three main steps. Okay. First step is called as selection of parents. The first step is called as selection of parents. So you select parents here in the first step and they are supposed to be pure line. I'll tell you what is a pure line right now. Okay. Second thing is called as crossing of parents to get F1. So you take the parents, you cross the parents and we get the F1 plants. And the third and the final step is called as selfing of F1. It is called as selfing of F1. F1 sigatala, you take the F1 plants and you cross them, and that's what is the final step. Three main steps in your hybridization experiments, and the same three steps are used in your monohybrid, dihybrid cross also. Okay. Now students, a true breeding line is one that has undergone continuous self-pollination. So I have a plant, I will make it undergo self-pollination until I get a pure breeding line. When I say pure line, it is supposed to express only one type of phenotypic expression. Sarna? Shows a stable trait, inheritance and expression of several generations. For generation it will express. So Mendel took 14 true breeding pea varieties. 14 true breeding pea varieties. Each one of them is expressing a particular trait. Okay. As a pair were similar except for one character with a contrasting trait. See, contrasting trait is there. Example nodi. For example, see here. Character, I told you. Character. Seed shape, seed color, flower color, pod shape, pod color, flower position and stem height. All of these are called as your character. Now, is the seed shape round wrinkled? Both of these are called as trait. They're called as trait. Now, one is called as a dominant trait, one is called as a recessive trait. Dominant trait is a trait which can be expressed all the time. It doesn't matter if the recessive trait is present. It doesn't care if recessive allele is present. Dominant trait is always expressed. Okay. Recessive trait is only expressed when dominant trait is missing. Okay. Now, color yellow green, flower color violet and white, Pod shape full and constricted. Pod color green and yellow, right? Flower position axial terminal. Stem height tall and dwarf. Example ಕೇಳಿದಾರೆ there was a PYQ in your KCET. KCET li ಕೇಳಿದ್ರು ಇದನ್ನ ಸರಿನಾ? They asked you for example, this was the one, which of the following is not a character chosen by the Mendel. Aata question direct. So it's a very simple thing. Remember the entire table. All of you should remember dominant one is more important to remember because mainly the questions come from the dominant trait. Okay. Now students, you might ask me, why sir? He could have taken rose plant, he could have taken a sunflower plant, he could have taken a green gram plant, some black gram plant. Why only pea plant? Because there are certain advantages for a pea plant. What are these advantages? Understand. For example, pure variety is easily available. Easily we can do pure lines. Large number of seed produced per plant. One plant is producing a lot of seeds. I can take a lot of seeds and plant more plants. Yes. Next thing is easily detectable contrasting characters. Pod shape, pod color, flower shape, flower color, stem height, seed color, seed shape. Everything is very properly visible. Okay. Can be crossbred manually. That is, emasculation can be done very easily here. Short lifespan, reproduce in 3 months. So every 3 months he can do a fresh experiment. For 7 years he did the experiment. So every 3 years he can start a new batch. Okay. Now, can be grown easily. Tumba fertilizer, tumba en bekagilla. No, very resistant also to pest. It can grow easily. So these are the main reasons why Mendel took the pea plant as his test subject. Now students, if you watched the video till now, quickly smash that like button right now and move ahead. Okay. Now students, next thing, by crossing, okay, so law of governs that inheritance takes place. There are 3 main laws which he gave. I already told you, that is law of dominance, law of segregation, and law of independent assortment. These are the 3 main laws which were given by Gregor Mendel. And out of these 3 laws, law of dominance and law of segregation, these 2 laws were given from monohybrid cross, while your law of independent assortment, this law right here, was given by dihybrid cross. Remember that much, because when we learn about monohybrid, when we learn about dihybrid, this will come again. Okay. Now students, now first we will go about inheritance of one gene, that is also called as your monohybrid cross. Can we start students? Can we start? All of you, a small water break. All of you drink some water. I'll give you 20 seconds. Go. 20 seconds. Water break. I want everyone to drink some water. Done? Okay. I believe all of you drank some water now and ready for monohybrid cross. Now I will write the entire monohybrid cross for all of you so you can learn better. Okay. Understand all of you. Monohybrid cross. Right? Monohybrid cross. Monohybrid cross. Understand students. Understand the monohybrid cross. Very, very important with respect to examination, because many questions will come from monohybrid, especially numericals will come here. I'm very sure you want to learn the examples, you want to learn the questions that will be coming up on the channel very soon, where I'll make you solve the questions from PYQ of every single chapter of KCET. That is coming up, don't worry. Now first thing is selection of parents, right? First step is selection of parents. So we are taking 2 types of parents here. One is homozygous dominant. One is homozygous dominant. Now what is the meaning of, first of all, homozygous? Understand. ನೀವು ಕಿಟ್ಕ್ಯಾಟ್ ಅಡ್ವರ್ಟೈಸ್ಮೆಂಟ್ ನೋಡಿದ್ದೀರಾ ಅಲ್ಲ? ಅದು 5 ಸ್ಟಾರ್ ಅನ್ಕೋತೀನಿ. 5 ಸ್ಟಾರ್ ಅಲ್ಲಿ we have Ramesh and Suresh. Ramesh and Suresh are twins. Ramesh and Suresh are twins. But students, do the twins look alike there? No. But do you have twins in the world which look completely alike, called as identical twins? They are like your identical twins. Homozygous condition is a condition in which 2 alleles are same to same. Now what is the allele now? Allele is an alternative form of a gene. One gene then call it. One gene, for example, let's say there is a gene for tallness. Gene for tallness. tallness के we have a gene here. Now this gene right here can have two alleles, alternative form, capital T or small t, small t. This is dominant allele, this is recessive allele. So a gene will have two alternative forms, like how we have two twins, no? Two twins, dominant twins, recessive twins. Dominant allele, recessive allele. They're present in pair always, present in pair. Okay, now understand students, we'll come back here. So first one is called as your homozygous dominant. So homozygous dominant, let's say capital T, capital T. This is for tallness. This is exact. This is tall. Homozygous dominant. Okay. The other one is homozygous recessive. Homozygous recessive. Understand students. Homozygous dominant is capital T, capital T. Homozygous recessive is going to be small t, small t. And this is going to be tall, this is going to be dwarf. It's going to be dwarf in nature. Dwarf. Homozygous dominant, homozygous recessive. These are basically, this is the selection of parents. Selection of parents. Now next step is something called as formation of gametes. Formation of gametes. Now from here, from this two allele, only one will go into the gamete, that is going to be your capital T. From here we will get one allele into a gamete, a small t. Remember, only one will go. Okay, so capital T, small t. Next step is called as F1. Formation of F1 or crossing over or crossing. So this T and this T will cross and we will get capital T, small t. This is called as the F1 generation, also called as the first filial generation. F1 generation or first filial generation. F1 generation. Okay. Now students, tell me, here we have capital T from tallness, small t from dwarfness. Now two alleles are present together. For one organism. And when two alleles are different now, here homozygous means same. Homozygous means same. This right here is now heterozygous condition. Heterozygous. This right here is called as your heterozygous. Heterozygous condition. Heterozygous condition. In heterozygous condition, now tell me students, capital T is more dominant. Yes. So this is going to be heterozygous dominant, and the trait which is observed here, is tall. This plant is going to be tall. So the F1 is going to be tall, heterozygous dominant, capital T, small t. The next step is called as selfing of F1. Okay. One second, students. Next step is called as your selfing of F1. Selfing of F1. So what do we have in F1, students? In F1 we had capital T small t crossing with capital T small t. Okay. This is your F1 right here. It is F1. Now again we need to form the gametes here. Yes, we need to form the gametes. For example, from here we will have capital T, small t gametes. From here we will have capital T, small t gametes. Okay. These are two different gametes we'll get here. Here we had homozygous condition, so only one gamete. Homozygous condition. Heterozygous condition, two separate gametes separately. Now the last step here is called as your selfing of F1. Now we need to do a square. Follow a separate method called as Punnett square method. This right here is called as your Punnett square method. Male, female. One is going to be male in this. One is going to be male. One is going to be female, obviously. Sexual reproduction. Okay. Now what do we have here? The gametes we have here is capital T, small t from one, capital T and small t from another. Now we do the cross. Capital T. Okay. Capital T, small t. Okay. Capital T, small t. And always write the capital T first, then small t. Dominant should be first, recessive should be second. Last one is small t, small t. Okay, so these are the different types of offsprings which we get after F1, and this entire offsprings are called as your F2 generation. They are called as your F2 generation. Second filial generation. That is, after the first crossing over, these are called as the second filial generation, also called as your F2 generation. Now, the last step here is to find the genotypic ratio and phenotypic ratio. But first, understand what is this meaning of phenotypic and genotypic. A phenotypic ratio is a ratio which tells you how an organism looks from the outside. That is called as a phenotype. For example, if I am tall, I will have capital T, capital T. Okay, so based on the alleles here, I can tell how a plant looks from the outside. That is called as phenotype. And how a plant looks from the inside, it means what is the genetic composition, what is the genes inside, that becomes a genotype. So phenotype from the outside, genotype from the inside. So phenotypic ratio means number of similar group of phenotype. I'll tell you an example, understand. Here we have, let's take phenotypic ratio first. What is the phenotypic ratio? Okay, phenotypic ratio. This is going to be tall. This is going to be tall. Why? Because capital T is dominant. This also is going to be tall, but this is going to be dwarf. Dwarf. So how many are tall, students? Tell me here. 1, 2, 3. 3 are going to be completely tall here. 3. 3 is to 1. 1 is going to be dwarf. That is called as your phenotypic ratio. Phenotypic ratio. Next thing is called as genotypic ratio. Genotypic ratio. Okay. In genotypic ratio, we look at the genotype. How many of them are homozygous dominant? How many of them are homozygous recessive? And how many offsprings are heterozygous dominant or recessive? Okay. Heterozygous 90 almost all the time it is dominant itself. Okay. Now see here. This is going to be homozygous dominant 1. Heterozygous dominant, heterozygous, right? Capital T, small t. Heterozygous. Capital T, small t, capital T, small t. Heterozygous dominant 2 are there. And homozygous recessive only 1 is there. So what is the genotypic ratio? Genotypic ratio is going to be 1:2:1 is going to be the genotypic ratio. And what is going to be the phenotypic ratio? 3:1 is going to be the phenotypic ratio, and this is called as your monohybrid cross. Done? Yes or no, students? Yes or no? All of you understood? This is called as your monohybrid cross. Okay. Now I'll move aside. I want everyone to write this down also one more time. I want everyone to write this down in your book right now and learn right now itself. What is the monohybrid cross? Now in examination, in your board examination, they can ask you directly to write the entire flow chart. You should be able to write the entire thing. Done, students? Done? Okay. Now let's see what is given you NCERT. Same thing. Tall dwarf, F1 generation we get, right? We take F1, we have all tall only. In F2 generation, we will have tall and 1 dwarf, right? 3:1 ratio. 3:1 ratio. Now let's see what is given your NCERT also. NCERT diagram missing. Missing andre missing ಈಗ ಏನು ಮಾಡಕ್ಕಾಗುತ್ತೆ ಹೇಳಿ. NCERT diagram missing ಇಲ್ಲ, ಇಲ್ಲಿದೆ ನೋಡಿ. ಓ ಇಲ್ಲಿ NCERT diagram. See here. Capital T, small t. So we are homozygous dominant, homozygous recessive. Okay. We're crossing over. Then we doing, this is your F1. This right here is, this is your F1. We get F1 here. We get the F1 here. F1. That is capital T, small t. Homozygous, heterozygous dominant. Now here we have selfing of F1. Selfing of F1. Capital T, small t, capital T, small t. And then what do we get? Phenotypic ratio 3:1. Genotypic ratio 1:2:1. That is going to be the genotypic ratio on the board. That is called as your monohybrid cross. And students, in the monohybrid cross, we took only a single character, that is your stem height. We did not take any other character. We took a single character, stem height, and we solved the entire equation, entire this thing. Okay, single. Now, students, this entire thing is called as Punnett. So I told you this is your Punnett. Okay, his name is only Punnett. R. Punnett. R.C. Punnett is the name. He was a scientist who discovered the Punnett square, who discovered the entire Punnett square. Okay. Now, students, inheritance of one gene. We learned here. Based on the observations, Mendel proposed that something was being stably passed down. All of you know, unchanged from parent to the offspring through gametes over successive generation. He called these things as factors. See, now we call it as gene. We know genes are passed. I know for a fact that we call genetics. From father to son, from father to daughter, from mother to son, mother to daughter. Genes are being passed. But that time, Mendel called it as factors. Okay. So now we call them as genes. Okay. Therefore, genes are a unit of inheritance. They contain the information that is required to express a particular trait in an organism. So all the information, how I talk, how I walk, how I say things, what is my, if I have any diseases, everything, all the information is stored in the genes itself. So everything is stored in the genes. Now, students. So where are genes? If this is the chromosome, all of you in the chromosomes, on the chromosome there is a segment where we have genes. For here we have stem height, here we could have seed shape, pod color. All of this information are present on the chromosomes, which are present on the genes. So genes are present on the chromosomes. Okay. So alleles can be similar in case, that is homozygous, I told you, capital T, or small t, homozygous, or they can be different, that is capital T, small t, that is heterozygous condition. This is the entire monohybrid cross right here. Okay, monohybrid cross right here. Now, students, after the monohybrid cross, now there is something called as test cross. Understand why test cross is done, what is a test cross, what are the different types of test cross. Understand, students. Before you understand test cross, I want everyone to understand something called as back cross. First of all, back cross. Back cross, there are two types of back crosses. Okay, two types of back crosses. In the first type, I take F1 progeny and cross it with dominant parents. Okay, dominant parents. The second type of back crosses, when I take F1 progeny and I cross it with recessive parents. Okay, such a type of cross is called as your outcross. This right here is called as your outcross. Not given your syllabus. This you do not have in your syllabus. Outcross is not there. When you take F1 progeny, not really. Here, in this entire cross, we learned here, right? You take the F1 progeny. Now, in monohybrid, what are you doing? In monohybrid, you're taking F1 and selfing of F1 you're doing. But in a test cross, you take the F1 and you cross it with, this is crossing with recessive parents. So, if this we are taking capital T, small t, and we're crossing with small t, that becomes a test cross. So, F1 plus recessive parents. For example, capital T, small t is crossing with small t. Such a cross is called as your test cross. And this is in your syllabus. Test cross is present in your syllabus. So all of you, we need to learn test cross. Now next thing is, I told you what is test cross now. Okay, I told you what is a test cross. Test cross is a cross. Test cross, students. Test cross is a cross between. It is a cross between F1 offspring and F1 offspring and F1 offspring with recessive parents. With recessive parent. Now you might ask me, sir, why are we doing it? What is the main need for test cross? And students, remember, every single time you do a test, why? ನೀವು exam ಯಾಕೆ ಬರೀತೀರಾ? To know, if to check your knowledge. To check your knowledge, you do it. Also to get a good marks or good life, basically. Okay. But why are we doing a test here? To find something. And what are we trying to find here? To know the genotype of F1. Adenge, how can we know genotype of F1? Let me tell you, students. This right here is a diagram given in your NCERT. All of you are aware of this diagram. Okay, all of you are aware of this diagram. Now here, I have a F1 plant. Here. This is your F1. This right here is your F1 plant. Now this F1 plant which we take has to be a dominant phenotype. It has to be a dominant phenotype, but unknown genotype. So why are we doing test cross? We are doing test cross to find the unknown genotype here. Okay. Now, if it is a dominant phenotype, what are the possible genotype? The possible genotype can be capital W, or capital W, small w. Yes or no? Both of them will give you purple flower, or violet flower here, Similarly, in NCERT they have given example. For example, let me just check what is given in your NCERT. In your NCERT they have given you, let's take, we have a tall plant. In F1. Tell me, what are the possible genotype? The possible genotype of a tall plant can be, capital T, it is completely dominant, or it can be capital T, small t. So all of you are understanding now. The genotype of a tall plant can be homozygous dominant, or it can be heterozygous dominant. Both the possibilities are true, but we do not know which one is it. To find the genotype of unknown genotype of F1, we do the test cross. See here. Here also we are doing it. Okay. We are doing to know the genotype of F1. So this is your F1. The possible genotype, it can be capital W, or it can be capital W, small w. Two possible things which can be there for the color of the violet flower here. Okay, violet flower. Now students, now. How do we do it? Okay, we do something called as test cross. In test cross, we take the F1. And we cross it with recessive parents. F1 with recessive parents. So homozygous recessive. Now students, we are trying to find the genotype of your F1 by looking at the phenotype of F2. Very important line. I'll write here for you in your box. Very important line if you want to understand test cross. Very important line for all of you. We are trying to find, okay, we are trying to find genotype of F1. Okay, F1 using or by identifying the F2 phenotype based on, students understand, based on how the plant is looking at F2. Plant nodake hengide acheಯಿಂದ. By looking at how it looks in F2, we will find the genotype of F1. So phenotype ಇಂದ genotype ಸಿಗ್ತದೆ ನಮಗೆ. ಹೆಂಗೆ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. Here. Now, there are two possibilities I told you. It can be capital W, or it can be capital W, small w. Now we do a cross. Here we get capital W, small w is the gametes. One here. So when we cross with recessive parents, which is small w, small. Okay. So what do we get? Capital W, small w, capital W, small w. This capital is coming from here. Small. This is the first case. This is the phenotype of your F2. Okay. Here, what was the phenotype of F2? This is the phenotype of F2 here. All of them are violet. Now, just because all of them are violet here, can I tell every single one of them will have one capital W for sure, which is dominant here, and henceforth this is the phenotype, this is the genotype. Now here, if I get two white flowers, it means that somewhere there was a small w in the genotype of F1. Somewhere in the genotype of F1. So we are trying to find the genotype of your F1 by looking at the F2 phenotype. If it is white color is possible, white color is coming in your F2, it means that somewhere there is a small w in F1. If you are getting all violet here, it means that there is always capital W in the F1. So with the help of your F2 phenotype, we are finding the genotype of your F1. That is called as your test cross. Okay. So I believe all of you understood till here. Now I told you already, from monohybrid cross we are deriving two laws, that is law of dominance as well as law of segregation. Now it is a time to understand these two laws. What is law of dominance and what is law of segregation? Ready? All of you ready, students? Trying to understand? Let's understand now. Yes. Give me a minute. Let me drink some water now. Now students, based on his observations on monohybrid cross, Mendel proposed two general rules of consolidate to his understanding of inheritance in monohybrid cross. Okay. Today these laws are called as principles or laws of inheritance. The first one is called as law of dominance. Second one is called as law of segregation. Understand. Each one of them is very important. The first one is called as your law of dominance. Law of dominance says that when there are two alleles, two contrasting alleles, capital T, small t, one will be dominant, one will be recessive. And the dominant allele will always express itself even in the presence of recessive. And the recessive can only express when the dominant is missing. Okay, see here. Characters are controlled by discrete unit called as factors. All of you know that. All of this information is transferred, entire thing is controlled by genes, and genes are nothing but factors here. Factors occur in pair, that is alleles, dominant, recessive. Okay. Genes also occur in pair. In a dissimilar pair of factors, capital T, small t, dissimilar pair of factors, one member of the pair dominates the other. This is dominating this one. Law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1. Yes or no? In the F1, we got capital T, small t, okay, in F1, and only capital T was expressing. Why? Because this was dominant. An expression of both in F2. In F2, both are expressing. Okay, that is small t was expressing. Okay, it also explains the proportion 3:1. How we get 3 tall, 1 dwarf. Wherever we have capital T, it is tall. Capital T, small t is also tall. Small t is dwarf. This small t is able to express itself. Why? Because capital T is absent there. That's why it's gonna be expressing. That is called as your law of dominance. Then what is law of segregation? Understand. Now students, this law of dominance is not universal. Law of dominance has certain exceptions. Incomplete dominance, co-dominance, exceptions are there. But your law of segregation is universal. It is universal. There are no exception to your law of segregation. That is, this here is universal. It's going to be universal. The law is based on the fact that alleles do not show any blending. This is the most important line here. The alleles do not show blending. I'll give you a simple example. ನೀವೆಲ್ಲರೂ ಓರಿಯೋ ಮಿಲ್ಕ್ ಶೇಕ್ ಮಾಡಿದ್ದೀರಾ ಮನೆಯಲ್ಲಿ ಯಾವತ್ತಾದರೂ? ಇಲ್ಲ ಓರಿಯಂ ಮಿಲ್ಕ್ ಶೇಕ್ ಮರ್ತೋಗಿ, ಯಾರು ಜ್ಯೂಸ್ ಮಾಡಿದ್ದೀರಾ, ಪಾನಕ ಮಾಡಿದ್ದೀರಾ, ಸಿಂಪಲ್ ಲೆಮನ್ ಜ್ಯೂಸ್ ಮಾಡಿರ್ತೀರಾ ಮನೆಯಲ್ಲಿ, ಸರಿನಾ? ಸೊ ಏನು ಮಾಡ್ತೀರಾ ಅವಾಗ ನೀವು, ಯು ವಿಲ್ ಟೇಕ್ 1 glass. In that 1 glass you add sugar, salt, lemon, water. ನೈಸ್ಲಿ ಮಿಕ್ಸ್. ಓಕೆ, ಮಿಕ್ಸ್ ಮಾಡಿದ್ವಿ. ನಾವು ಸ್ಟೂಡೆಂಟ್ಸ್ ಎಲ್ಲ ಮಿಕ್ಸ್ ಮಾಡಿದ್ಮೇಲೆ ಕ್ಯಾನ್ ಯು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ lemon juice out of it? Can you extract water out of it? Can you extract salt and sugar separately? Very easy to. Can you do it at home? Tell me, can you do it at home? Not possible. Because all of them have shown blending now. ಎಲ್ಲ ಮಿಕ್ಸ್ ಆಗೋಗಿದೆ, ಎಲ್ಲ ಮಿಕ್ಸ್ ಆಗೋಗಿದೆ. But here alleles mix ಆಗಲ್ಲ ಆ ಥರ. When I say capital T small t are present here, capital T small t are present here, it doesn't mean just because small t is not expressing, it doesn't mean capital T small t ಎರಡು ಮಿಕ್ಸ್ ಆಗ್ಬಿಟ್ಟಿದೆ and only capital T is present and it is expressing. ಆ ಥರ ಆಗಲ್ಲ. So alleles do not show blending. ಯಾವತ್ತು when we have capital T small t are there, they are not blending, they're separate. They're separate. I'll tell you an example. One proper example is there. Okay, that both the characters are recovered as such in F2 generation. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ ಸ್ಟೂಡೆಂಟ್ಸ್. If there was blending, ಅಕಸ್ಮಾತ್ ಇಲ್ಲಿ ಬ್ಲೆಂಡಿಂಗ್ ಆಗ್ತಾ ಇದ್ರೆ, see this, we'll go back to your monohybrid. Now students, here we have capital T small t. ಅಕಸ್ಮಾತ್ ಇಲ್ಲಿ ಬ್ಲೆಂಡಿಂಗ್ ಆಗ್ತಾ ಇದ್ರೆ, if there was blending, then how can we get small t again? How is the small t even coming up again? Not possible, no. So the scientist, that is Gregor Mendel, is saying alleles do not blend, they segregate separately during gamete formation. Artha ಮಾಡ್ಕೊಳ್ಳಿ, very important line. That is, alleles show, when two alleles are present together, they do not show blending. That is, they do not show blending, they will segregate separately during gamete formation. Nodi illi, during gamete formation, what we have here? During gamete formation, we had your capital T separate, small t separate. Yes or no? That's why it is called as law of purity of gametes. Koda kariti ನಾವು ಇದನ್ನ law of segregation. That is, alleles segregate during gamete formation. And here each gamete is pure for one trait. This gamete is pure for tallness, this gamete is pure for dwarfness. So law of purity of gametes and also law of segregation. Alleles segregate during gamete formation. Okay, during gamete formation. It is universal. This right here is said to be universal in nature. Law of segregation, no? Law of segregation. Though the parents contain two alleles during gamete formation, the factors or alleles of pair of segregate from each other such that gamete receives only one of the two factors. Capital T small t, gamete is receiving one of the two factors. This gamete is receiving one factor, this gamete is receiving another factor. Of course, a homozygous parent produces all gametes that are similar, while heterozygous produces two kinds. Heterozygous producing two kinds, each having one allele only. Students, if you want these notes, you should join the Telegram channel. That is, we Karnataka Telegram channel. Description there is a link. Click on the link, join the Telegram channel, download the notes and enjoy and study hard. Okay, and if you want to join my Telegram channel where I post exclusive videos, where I talk about plans, where I talk about your NEET preparation, when I talk about your preparation of KCET, this is my separate channel, my Telegram channel, Basivaraj Sir Biology. Okay, Basivaraj Sir Biology. This is my Telegram. Okay, so there are more than if I remember, I didn't check recently, there were around 2,500 students are there. More than 2,500 students are already there in Telegram channel. So go quickly join right now. Okay, that is my Telegram channel. Now students, that is your entire law of segregation. Now students, next concept is called as your incomplete dominance. See, all of you are aware, I told you already, law of dominance has certain exceptions. One such exception is called as incomplete dominance. That is, ಇದು ಡಾಮಿನೆಂಟ್ ಅಲ್ಲೀಲು ಕೆಲಸ ಸರಿಯಾಗಿ ಮಾಡಲ್ಲ. ಇಟ್ ಇಸ್ ನಾಟ್ ವೆರಿ ಡಾಮಿನೆಂಟ್ ಆಗಿ ಕೆಲಸನೇ ಮಾಡಲ್ಲ ಅದು. And because of that, your recessive allele is there, no? Your recessive allele is also expressing. ನಾನು ಸ್ವಲ್ಪ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡ್ತೀನಿ ಅಂತ. So no. So what is happening in incomplete dominance is the dominant allele is incompletely dominant, and because of that, the recessive allele is also expressing. It is also expressing in nature. Understand now? Okay. See here. When experiments on pea plant were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the parents. ಏನಿದು ಎರಡು ಪೇರೆಂಟ್ ತರನೇ ಇಲ್ಲ. One parent is red, one parent is white, but the offspring is pink. One parent, male parent, let's say red, female parent is white. Child is pink color. I think Bandhu, that is because of incomplete dominance. Okay. That is inheritance of flower color in dog flower, that is snapdragon or atrium species, is a good example to understand the incomplete dominance. Start madada artha madkono. Okay. Can we start, students? Incomplete dominance now. Very important to understand incomplete dominance. I will write and teach you, students. I'll write and teach you. Understand incomplete dominance. Then I'll show you the entire graph, entire chart. Okay. The first step is parents. Selection of parents. Yes or no? First step is selection of parents. Let's take your let's take one is red flower, one is red flower crossing with white flower. White flower. One is going to be homozygous dominant. Let's say capital R, capital R. One is going to be homozygous recessive. Small r, small r. This is going to be red. This is going to be white. Next step is to form the gametes. Next step is gamete formation. This is gamete formation. Okay. Now what are the gametes do we get here? From here we get a single gamete, capital R. Here we get small r. Okay. Next, this is your F1 generation. This here. Next we do crossing. That is your formation of F1. Cross it. And we get capital R, small r. Now students, what is exactly happening here is, in your normal monohybrid, this was supposed to be red. This was supposed to be red in normal monohybrid. But right now, this R right here, this R right here is incompletely dominant over small r. So the offspring here which we get, the offspring which we get here is said to be pink flower. It's pink flower. We get pink flower here. Pink flower. Okay, pink flower. Now next step is called as what? Selfing of F1. Selfing of F1. Selfing of F1. So what do we do right now? We take capital R, small r crossing with capital R, small r. We form gametes. Two gametes, capital R, small r, capital R, and small r. These are different type of gametes we get here. Okay. Now let's draw Punnett square here. Let's draw Punnett square. Let's draw Punnett square. Okay. Male, one is female. Capital R, small r, capital R, small r. From one parent, capital R, small r, capital R, small r. Okay. Now we write the gametes. Capital R, capital R, capital R, small r, capital R, small r, small r, small r. Now understand, students, understand all of you. This is going to be completely red. This is going to be pink. Why? Because this is incompletely dominant over the other one. This also is going to be pink. And this is going to be completely white. It's going to be completely white because it is completely recessive in nature. Recessive in nature. Now, what is the phenotypic and genotypic ratio here? Understand. What is the phenotypic and genotypic ratio here? What is the phenotypic and genotypic ratio right here? Now, if you see here, one is red, red. Two pink, two pink. And one white, one white. 1:2:1, 1:2:1. Now let's take homozygous dominant, one. Heterozygous dominance, two are the heterozygous condition. Heterozygous condition, there are two are heterozygous condition. Homozygous recessive, one. So what is the phenotypic ratio and genotypic ratio? Both are 1:2:1. And here can I tell your phenotypic ratio is equal to genotypic ratio. Equal. Just like the test cross. Here also we have phenotypic ratio is equal to genotypic ratio. And this is called as your incomplete dominance, where one allele is incompletely dominant over the other allele. Okay, see here. It's given you an NCERT also. Red, white, gametes, F1, all pink. Selfing of F1, video. Then we get red, pink, white, 1:2:1. Genotypic, 1:2:1 is genotypic ratio. Both are equal here, right here. Okay. So what happened? So here we learned that one is incompletely dominant over the other one. Okay. Clear, all of you clear? Yes. Easy. Now students, there is something called as concept of dominance. Concept of dominance. Why one is dominant, why one is recessive? The answer is very simple. Because the recessive one has undergone certain mutation or changes. Because of it, it is not able to produce the actual expression. That is mutation. It has undergone mutation. In your NCERT, they are giving you an example of an enzyme. NCERT, they are giving you an example of an enzyme, like a modified allele. Recessive one is also called as modified allele. Modified allele, then enzyme, one enzyme can produce normal, less efficient. Non-functioning enzyme, no enzyme at all. This is said to be dominant. This is recessive. This also is recessive in nature. So recessive allele, also called as modified allele, it's because of certain mutation or certain changes. That is why we get recessive alleles. Okay. Now, the next exception to the dominant allele, next exception to the principles of dominance. The next exception is called as codominance. So what exactly happens in your codominance, students? See, in your incomplete allele, incomplete allele, the F1 did not look like your mother or father. Did not look like any of the parents. But in codominance, it will look like both. Mother tarano irutte, father tarano irutte, ಎರಡು ತರನೇ ಇರುತ್ತೆ ಇದು. ನೋಡಿ ಇಲ್ಲಿ ಎರಡು ತರನೂ ಇರುತ್ತೆ. This is called as codominance. Why? Because the two alleles present here, both are dominant, both are expressing here. ನಾನು express ಮಾಡ್ತೀನಿ, ನೀನು express ಮಾಡು. There is nothing called as recessive allele only here. Nothing called as recessive allele at all here. So both are expressing, and that is called as your codominance. Now students, to understand codominance, you need to understand one more concept called as multiple allelism. ಏನಪ್ಪ ಇದು multiple allelism ಅಂದ್ರೆ, generally one allele has two alternative forms. Generally one allele has two alternative forms, all of you know. But in multiple allelism concept, one gene will have multiple alleles. ಎರಡು ಮಾತ್ರ ಅಲ್ಲ, 3, 4, 8, 10 ಆ ತರ ಇರುತ್ತೆ. Multiple alleles are there. Okay, that is called as multiple allelism. See here. And the example of multiple allelism, one of the examples is your ABO blood grouping. Example is ABO blood grouping. Now understand students, ABO blood grouping is a typical example here. Okay, now in ABO blood grouping we have three different alleles. In ABO blood grouping we have three different alleles here. Okay, let's say three different alleles. Three alleles are present here. We have your IA, IB and IO. We can just say IO, I small I or IO. Okay, three different alleles. Out of three different alleles, these two are dominant and this one is recessive. These two are dominant, this one is recessive. ಎರಡು ತರ ಹೆಂಗಾದ್ರೂ ಬರೀಬಹುದು ಇದನ್ನ. Okay, so three different alleles. Now you might ask me, students, ಈ ಮೂರು allele ಇದೆ, ಈ ಮೂರು allele ಕೂಡ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಇರುತ್ತಾ? ಸಾಧ್ಯ ಇಲ್ಲ. Why? Because we are diploid. How we have one chromosome coming from father, one chromosome coming from mother, in alleles also one comes from one parent, one comes from one more parent. So two alleles are only present in one organism. Okay, so even though we have three different alleles here, out of three only two will be present in our body. Only two of them will be present in our body. IA ಮತ್ತೆ small I ಇರಬಹುದು, capital IB or I ಇರಬಹುದು, ಇಲ್ಲ ಇದು ಎರಡು ಇರಬಹುದು. Those are possibilities. For example, students, for example, let's take a positive allele irutte, A allele irutte. A group alli there are possibilities are, we can have IA, two alleles, or we can have IA and small I. B positive, B allele irutte, IB, or it is going to be capital IB and small I. Possibilities, yes, it is a possibility. Okay, now what about AB blood grouping? In AB blood group in our room, we will have capital A and will be. Both dominant alleles are present. Okay. Now what about O blood grouping? In O blood grouping, we have do not have any of II. We will have I small I will be present here. That is basically O blood grouping. Now you might ask me, sir, ಯಾಕೆ ಈ ತರ difference ಬರುತ್ತೆ? The difference is basically the type of sugar which they have. For example, if you take O blood grouping, the antigen H do not have any sugar at all. Sugar illa, extra sugar illa illi. But if you look at your A blood grouping will have an additional N-acetyl glucosamine. Your B blood grouping will have an additional galactose here. Galactose is going to be, when they have AB blood grouping, you have galactose here and N-acetyl glucosamine. So both of the sugars will be present. That is what makes IA and IB different. Okay. Now, till now we have discussed there are F1 resembles either the parents. Here it resembles both the parents. Resembles both the parents. IA and IB are completely dominant over I. In other words, IA and I are present only in IA expression because I does not produce any sugar. I is not producing any sugar. It is recessive completely. Okay, it is recessive completely. Okay. So based on that, we have the table here. I told you, yavattu A blood grouping ಏನು ಇರುತ್ತೆ? IA ಇರುತ್ತೆ. AB ಗೆ IA IB. Other possibilities, IA and small I is a possibility. AB ಗೆ AB, B ಗೆ IB IB or IB small I, O ಗೆ II ಇರುತ್ತೆ. This is a table for your codominance human blood grouping. Human blood grouping. Okay. That is your multiple allelism. Now students, there is one more small concept given you in NCERT called as star synthesis. Okay. Star synthesis is an example of your pleiotropy and also your, so one gene affecting multiple expression, and also it is an example of your incomplete dominance. ಅರ್ಥ ಮಾಡ್ಬೋದು, ಸ್ಮಾಲ್ ಕಾನ್ಸೆಪ್ಟ್ ಇದು, ಕೇಳಲ್ಲ ಜಾಸ್ತಿ, ಕೇಳಲ್ಲ ಇದನ್ನ. ಇನ್ star synthesis what happens is, ನಿಮ್ಮ ಕ್ಲಾಸಲ್ಲಿ there are few students who are good at studies. There are few students who are good at sports. There are few students who are good at both also. But is there a possibility that there is one student who is good at sports, good at studies, who is also good at everything in the world? Athar agutta? Agala. Very rare, very rare. We can never find one person who is a top level Olympics player and also a top level genius. Okay, who is a top level maths professor or something. Athara tumba rare agadu. Very few possibilities that one of the student is good in everything in the world. Similarly, when one gene is producing multiple phenotypes, it doesn't mean that it is going to produce dominant phenotype all the time. One gene can produce a dominant phenotype for a seed shape, but it doesn't mean it will produce starch at the same time. Like how I told you, one student can be good at studies. It doesn't mean that he should be good at sports also same time. It doesn't mean that. Okay. So that is the example here. See here. Heterozygous produces round seed. Okay. And so B seems to be dominant allele. But a starch grain produced from the intermediate size. So it is producing round seed is producing, but it is not. It is producing intermediate starch grain. Starch grain produce ಮಾಡ್ತಾ ಇಲ್ಲ. So it is good at what? One gene is good at one thing, but it is very intermediate other thing. So it is incomplete dominant here. It is dominant here, incomplete dominant here. Okay. You can read this. So that is the end of your entire monohybrid cross. Now we will come to something called as dihybrid cross. Ready now students? Ready now all of you? Now we'll start something called as dihybrid cross, and I will write and teach you entire thing. I will write and teach you the entire thing now. Can we start students? Let's start your entire dihybrid cross now. Okay. It's also called as your inheritance of two genes. It is also called as inheritance of two genes. Now here, the first thing which you need to know, here is, in your monohybrid cross, we only had a single character. In your monohybrid cross. But in your dihybrid cross, we will study right now. In dihybrid cross, we have two different characters here. There we had only stem height, tall, short. Stem height was there, tall, short was there. But right now we have seed shape, seed color. Seed shape can be round and wrinkled. Seed color can be green and yellow. So two different characters are taken here. So first thing is parents. First step is parents. For here we have capital R, capital R, capital Y, capital Y. Now what is this? This capital R, capital R, capital R, capital Y tells you R represents round, dominant, seed shape, yellow, dominant, seed color. So here we have seed shape and seed color. Seed shape and seed color. Seed shape and seed color both are dominant here. Both are said to be dominant. Okay. Now what is the other parent? Other parent is small r, small y, small y. This right here is a recessive one. This is a recessive one. That is wrinkled green. Both are recessive traits. Here we have round, wrinkled, yellow, green. Okay. Now, next step is formation of gametes. Parents are to gametes. Okay. Now here, students, we will have two different gametes here. Here we will have capital R, capital Y. Okay, here capital R, capital Y. Each one is representing only a single character. In this, we have two different gametes, but each one is representing only one of the character. This is representing seed shape. This is representing seed color. Remember, contrasting traits are not present together. Okay. Now, here. Small r, small y. Small. Next step is called as F1. We crossover. We get capital R, capital Y, small y. Okay. Now here, this is going to be capital R is dominant, so round. Y is dominant, so we get a yellow. So we will get round and yellow right here. We'll get round and yellow right here. Round and yellow. We'll get round and yellow right here. Round and yellow. Now next step is called as selfing of F1. Next step is called as, this is F1. This right here is the F1. Next step is called as selfing of F1. Selfing of F1. To find selfing of F1, students, first you need to find the gametes, right? So first you need to find the gametes. Capital R, capital Y, small y. So how do you find the gametes here for this? Let me teach you. We have capital R, capital Y, small y. Simple way to find the gametes here. A small hack for all of you. Okay. Multiply this with this and this with this. This with this and this with this. Simple hack. Okay. So what do we get here? The gametes which we get from one of the parent is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So 4 different gametes from one parent. And we are crossing with this with other parent also. It's selfing of F1. This is selfing of F1, right? Selfing of F1. It is selfing of F1. So what do we do? Here also we'll get 4 gametes. Now again we do something called as Punnett square. We do something called as your Punnett square. Okay. Male, female. We'll write one is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Same thing right here also. Capital R, capital Y, capital R, small y, small r, capital Y, small r, capital Y, and small r, small y. Okay. Now we need to fill the entire table now. Entire table will fill, and I'm filling right now. I want everyone to fill also with me. Okay. Can we start? Yes, sir. Let's try to fill it up. First we have what? Capital R, capital R, capital Y, capital Y. What will it be? Round yellow. Yes or no? It's going to be round and yellow. This is going to be round and yellow. Round and yellow. Okay, first one is going to be round and yellow. Now let's fill it up first. Capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital R, small r, capital Y, small y. Here what do we get? Capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y, capital R, small y, small y. Correct, no? Capital R, small r, small r. Okay. Capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, small y, small y. That is your entire table right here. Now students, how many of you are round yellow? Let's count. See, this is going to be round and yellow. Round and yellow. Round and yellow. This is going to be round yellow. Round yellow. Round and yellow. Capital R, capital Y. Capital Y, round and yellow. Round and what is small y, students? Tell me what is small y here? This is going to be round, but this is going to be green. Round, green, round and green. Round and green, because small y is there. Okay, so next step is round and green. Let's write here also. Round and green. Okay, round and green. Okay. Now, capital R, capital Y, round and yellow, again same thing. This is going to be what? Round and green again. Round and green. Round and green. Let's mark it. Okay. Capital R is capital Y, round and yellow, again same thing. Round and yellow. Now this is going to be small r. It means wrinkled yellow. Wrinkled yellow. Okay. Let's use red, pink for this. Wrinkled yellow. Wrinkled yellow. Okay. Again, wrinkled yellow. Wrinkled yellow. Okay. I can see one more here, wrinkled yellow. Okay. This is going to be round and yellow. Round and green. Round, small y is green. Wrinkled yellow. Last one is going to be wrinkled and green also. It's going to be, make a star for this one. It's going to be wrinkled and green. Small r, wrinkled. Small y, green. Wrinkled, green. Wrinkled green. Okay. Now let's count how many are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them. 1, 2, 3. Wrinkled yellow. 1, 2, 3. Wrinkled green. Only 1. So what is the phenotypic ratio here? How it looks from the outside? The phenotypic ratio of your dihybrid cross is going to be 9:3:3:1. That is going to be your phenotypic ratio in your dihybrid cross right here on the board with the help of a beautiful table. Now students, in examination they will never ask you what is the dihybrid cross. Not possible. But I'll tell you a small trick to remember dihybrid cross right now. Okay. Now I'll tell you a small trick which I usually tell. For example, tell me students, let's take about phenotypic right now. Phenotypic ratio. Phenotypic. Now all of you are aware, right? Dihybrid cross is technically two characters. It is twice the amount of monohybrid. Twice the amount of monohybrid. Two monohybrid, one dihybrid. Two monohybrid, one dihybrid. Tell me, what is the phenotypic ratio in monohybrid? 3:1. 3 tall, 1 dwarf into 3 tall, 1 dwarf. 3 3s are 9. 3 into 1 is 3. 1 into 3 is 3. 1 into 1 is 1. 9 is 3:1 phenotypic ratio. What is the genotypic ratio? Genotypic ratio. Genotypic ratio of monohybrid was 1:2:1 monohybrid. 1:2:1 was the monohybrid. Cross it. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 2s is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. And 1 into 1 is 1. That is 1:2:1:2:4:2:1:1. That is your genotypic ratio of your dihybrid cross. Small trick will save you a lot of time in the future if you want to ever flex on your friends that you know genotypic ratio. Okay. Now that was your entire dihybrid cross. Entire dihybrid cross given your NCRT. This is your gamete formation. This is your gamete formation right here. Selfing of F1, F1, F2. We get F2 here. 9:3:3:1 is your genotypic ratio. Okay. Now students, with the help of your genotypic ratio, we get one law I told you. Genotypic ratio tells you the law that is called as law of independent assortment. Now what does law say? Understand. Okay. Mendel's law of independent assortment states that when two pair of traits that are combined in a hybrid, two pair of traits combined in a hybrid, one trait is going to be your one trait is going to be that is going to be round, one is going to be wrinkled here. Round and wrinkled are present together here. Okay, round and wrinkled are present together. In a hybrid, segregation of one pair of character, segregation of your seed color and segregation of seed shape, they will happen independently. There is no blending again. Segregation of seed color, that is your yellow and green. Segregation of seed shape, wrinkled and your round. Segregation of these two characters will happen independently. That is called as your law of segregation. That is called as law of segregation. Okay. Now I'll tell you one small sad story, student. Sad story. That is, even though all of this work was done by Mendel, students, all of this work was done by Mendel, but Mendel's work was never recognized. Mendel's work was never recognized until his very death. Okay. There are many reasons for it, and later on his work was identified. Who identified? All that I will tell you later on. Okay. Now understand, students. Mendel published his work on inheritance of characters in 1865, but several reasons it remained unrecognized till 1900s. So it was completely unrecognized. No one cared about his work. Okay. So first, why? First, communication was not easy that time as it is now. ಈಗ ನೀವು ಏನೇ ಹೊಸ ಡಿಸ್ಕವರಿ ಮಾಡ್ದೇನು ಮಾಡ್ತೀರಾ, ನೀವು ಇನ್ಸ್ಟಾಗ್ರಾಮ್ ಅಪ್ಲೋಡ್ ಮಾಡ್ತೀರಾ, ಯೂಟ್ಯೂಬ್ ಅಪ್ಲೋಡ್ ಮಾಡ್ತೀರಾ, ರೆಕಗ್ನಿಷನ್ ಬಂದ್ಬಿಡುತ್ತೆ, ವೈರಲ್ ಆಗಿ ಹೋಗುತ್ತೆ. ಬಟ್ ದಟ್ ಟೈಮ್ ನಥಿಂಗ್ ವಾಸ್ ದೇರ್. Okay. Secondly is concept of genes factors in Mendel's words as stable discrete units that control expression of traits and pair of alleles which do not show blending. ಈ ಎಲ್ಲ ಕಾನ್ಸೆಪ್ಟ್ ನಾವು ಅರ್ಥ ಮಾಡ್ಕೋತಾರಲ್ಲ ಯಾರು. ಆ ಕಾಲದಲ್ಲಿ ಎಲ್ಲರೂ ಬ್ಲೆಂಡಿಂಗ್ ಇಂದ ಬಿದ್ದಿದ್ರು. ಥಿಯರಿ ಆಫ್ ಸ್ಪೆಸಿಯೇಷನ್ ಆಗಲಿ, ಡಾರ್ವಿನ್ ಥಿಯರಿ ಆಗಲಿ ಬಿದ್ದಿತ್ತು. ಸೊ ಹಿಸ್ ಥಿಯರಿ ನ್ಯೂ ಥಿಯರಿ. ಯೋಚನೆ ಮಾಡಿ, ಸಡನ್ ಯಾರು ಬಂದ್ಬಿಟ್ಟು ನೀವೇ ಓದ್ತಾ ಇದ್ರಲ್ಲ ತಪ್ಪು, ನಾನು ಹೇಳ್ತಾ ಇರೋದು ಕರೆಕ್ಟು, ನಾನೇ ತುಂಬ ಬಾಸ್ ಇಲ್ಲಿ ಅಂತ ಹೇಳಿದ್ರೆ ಯಾರು ಇನ್ನು ಒಪ್ತಾರಾ? ಒಪ್ಪಲ್ಲ. ಹಂಗೆ ಆಯ್ತು ಮೆಂಡಲ್ ಕೂಡ. ಸರಿನಾ? Thirdly, Mendel's approach for using mathematics to explain biological phenomena was totally new and unacceptable to many biologists at the time. Accept ಮಾಡಿರಲಿಲ್ಲ. So his work was completely unrecognized until 1900s. No one even understood what he was doing that time. Okay. So finally, through Mendel's work suggested that factors genes were discrete units, he could not provide any physical proof for the existence of factors and that what were made up of. So physical proof irlilla. Okay, microscope illa time alli. I am telling there's a gene, but right now I can tell there's a gene because I can identify the gene. But that time, not possible because there is no microscope to it. There is no proof to show what is the gene. No proof. No one will care. In 1900s, three scientists, that is Deveries, Korns, and Schumacher independently rediscovered Mendel's result on the inheritance of characters. Also, by this time, due to the advancement in the microscopy that were taking place, scientists were able to carefully observe the cell division. So they observed cell division and they later identified that yes, Mendel was correct and he was correct all along. ಈ factors ಅಂತ genes ಇದೆಯಲ್ಲ, these genes are present on the chromosomes and we can see them segregating during cell division. Everything was finally came to light. That was Mendel's work. So students, until his death also, because of his kidney failure and everything, he could not see the amazing work he has done. But fortunately, we are talking about him right now. We are remembering Gregor Mendel. I hope he is, somewhere in the world, somewhere in the heaven, or he'll be able to know that yes, his work is being used right now, and because of his work, there are advancements in your genetics and everything, and that is why Gregor Mendel is called as your father of genetics. Father of genetics. Okay. Now, students, now I will teach you something called as chromosomal theory of inheritance. Very simple concept. The last concept for today's day. Today is not last concept. After this we will do, but this is the concept which bridges this chapter, half point of this chapter to the next chapter. Okay, that is called as your chromosomal theory of inheritance. Now, what does it tell you? Let's understand. Okay, now it's very simple. ಕೇಳೋದಕ್ಕೆ ತುಂಬಾ ಕಾಂಪ್ಲೆಕ್ಸ್ ಆಗಿದೆ. ಓ, chromosomal theory of inheritance ಅಂತ ಕಾಂಪ್ಲೆಕ್ಸ್ ಅಲ್ವೇ. It's very very simple. Chromosomal theory of inheritance tells you that the behavior of chromosome and behavior of a gene is same to same. It's same to same. ಯಾಕಂತ ಹೇಳ್ಕೊಳ್ಳಿ, ಯೋಚನೆ ಮಾಡಿ. Now students, if this is my hand, I am moving my hand from here to here. I'm moving my hand from here to here. Does my fingers also move along with it? And fingers still there. Fingers also came here. Obviously, fingers are present on the hand. Fingers are present on the hand. ಅಕಸ್ಮಾತ್ ನನ್ನ ಕೈ ಮೂವ್ ಆದ್ರೆ ನನ್ನ ಫಿಂಗರ್ಸ್ ಕೂಡ ಮೂವ್ ಆಗಲೇಬೇಕು. Or other example I'll give you. I'm standing here. I moved from here to here. ನಾನು ಮೂವ್ ಆಗ್ಬೇಕಾದ್ರೆ ನನ್ನ ಬ್ರೈನ್ ಕೂಡ ಮೂವ್ ಆಯ್ತಾ ನನ್ನ ಜೊತೆಗೆ? ಮೂವ್ ಆಯ್ತು ಸರ್. ಯಾಕೆ? ಬ್ರೈನ್ ನಿಂದ ಬಾಡಿ ಮೇಲೆ ಇದೆ, ಬ್ರೈನು ತಲೆಯಲ್ಲಿದೆ, ಬ್ರೈನು ನಾನು ಇದೆ ತಾನೆ. ಸೊ ನಾನು ಮೂವ್ ಆದ್ರೆ ಬ್ರೈನ್ ಕೂಡ ನನ್ನ ಜೊತೆಗೆ ಮೂವ್ ಆಯ್ತು. ಸೇಮ್ ಕಾನ್ಸೆಪ್ಟ್ ಯು ನೀಡ್ ಟು ಅಪ್ಲೈ ಫಾರ್ ಯೋರ್ chromosomal theory of inheritance. Chromosomal theory of inheritance say that the behavior of chromosome is similar to behavior of genes. ಅಕಸ್ಮಾತ್ ಕ್ರೋಮೋಸೋಮ್ ಮೂವ್ ಆದ್ರೆ ಜೀನ್ಸ್ ಕೂಡ ಮೂವ್ ಆಗುತ್ತೆ. ಕ್ರೋಮೋಸೋಮ್ ಇಸ್ ಸೆಗ್ರಿಗೇಟಿಂಗ್, ಜೀನ್ಸ್ ಕೂಡ ಸೆಗ್ರಿಗೇಟ್ ಆಗುತ್ತೆ. ಯಾಕೆ? ಲೈಕ್ ಹೌ ನನ್ನ ಬಾಡಿಯಲ್ಲಿ ನನ್ನ ಬಾಡಿ ಮೇಲೆ ಬ್ರೈನ್ ಇದೆಯಲ್ಲ, ಆ ತರ ಕ್ರೋಮೋಸೋಮ್ ಮೇಲೆ ಜೀನ್ಸ್ ಇದೆ. ಜೀನ್ ಇಸ್ ಈಕ್ವಲ್ ಟು ಬ್ರೈನ್, ಕ್ರೋಮೋಸೋಮ್ ಈಕ್ವಲ್ ಟು ಬಾಡಿ. ಇಫ್ ದ ಬಾಡಿ ಇಸ್ ಮೂವಿಂಗ್, brain is moving, chromosome is moving, gene also is moving, because genes are present on the chromosome. Okay, this led to discovery of structural genes in the nucleus that appear to be double and divide before cell division. They are called as chromosomes. Okay, the chromosome movement during meiosis had been worked out. And students, are quoted now. Chromosomal theory of inheritance was given by Walter Sutton and Theodore Boveri. Noted that behavior of chromosome was parallel to the behavior of genes, and they used chromosome movement to explain Mendel's law. Akasmat, if I can explain how the chromosomes are moving and segregating, I can also explain how genes are also moving. Okay, genes are also moving. Like how we get one chromosome from father, one chromosome from mother. Similarly, we get one allele from one parent, one more allele from other parent. Okay, same to same. See here, ಅಕಸ್ಮಾತ್ ಈ ಕ್ರೋಮೋಸೋಮ್ ಎರಡು ಮೂವ್ ಆಗ್ತಿದ್ರೆ, ಸಪರೇಟ್ ಆಗ್ತಿದ್ರೆ, ಅದ್ರ ಮೇಲೆ ಜೀನ್ಸ್ ಕೂಡ ಸಪರೇಟ್ ಆಗುತ್ತೆ. ಸರಿನಾ? The important thing to remember that the chromosomes are called as well as genes occur in pair. The two alleles of a gene pair are located on the chromosome, homologous sites or homologous chromosomes. So genes are present on the chromosome. ಇಲ್ಲೊಂದು ಕ್ವಿಕ್ ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟಿದ್ದಾರೆ ನಿಮಗೆ. ಲೈಕ್ ಇಫ್ chromosomes occur in pair, genes occur in pair. Chromosomes segregate at the time of gamete formation. ಆಗುತ್ತೆ. Chromosomes separate ಆಗುತ್ತಲ್ಲ? ಆಗುತ್ತೆ. Chromosomes ಆಗುತ್ತೆ. Genes ಕೂಡ separate ಆಗುತ್ತೆ. Especially in anaphase 1. Anaphase ಅಲ್ಲಿ separate ಆಗುತ್ತಲ್ಲ? Anaphase 1 ಅಲ್ಲಿ homologous chromosomes separate. Okay. Same thing is happening here. So both of them have some kind of relation. ನೋಡಿ. Anaphase 1 ಅಲ್ಲಿ homologous chromosomes separating. In anaphase 2, sister chromatin separating. Similarly, genes present there also separating. Okay. So Theodore Sutton and Boveri took the help of chromosome movement and also studied the principles of inheritance by given by Mendel. ಎರಡನ್ನ ಕಂಬೈನ್ ಮಾಡಿ we got the chromosomal theory of inheritance. ಎರಡನ್ನ ಕಂಬೈನ್ ಮಾಡಿ we got chromosomal theory of inheritance. But one missing factor ಇತ್ತು. Something is missing right now. ಏನು missing? Experimental proof missing ಇದೆ. And this experimental proof was given by none other than T.H. Morgan. T.H. Morgan was a scientist who worked on Drosophila. He came and told us, wait ಮಾಡಿ, ನಾನು ಹಿಂಗೆ proof ಕೊಡ್ತೀನಿ ಇಲ್ಲಿ. Okay, the proof was given by T.H. Morgan. Okay, experimental verification of chromosomal theory of inheritance by T.H. Morgan, Thomas Hunt Morgan and his colleagues led to the discovery of the basis of variation that sexual reproduction produced. Morgan worked on tiny fruit flies called as Drosophila melanogaster fruit fly. ನೋಡ ಇದು ಯಾವ ಥರ ನೋಡೋ ಗೊತ್ತಾ, ಚಿಕ್ಕದು ಬರುತ್ತಲ್ಲ ಡಾಂಬಾರಿ ಏನೋ ಅಂತಾರಲ್ಲ, ಅದನ್ನ ಅದು ಒಂದು ಬನಾನಾ ಪೀಲ್ ತೊಳಿ, ಆ ಬನಾನಾ ಪೀಲ್ನ ಬಿಸಾಕಿ ಆ ಕಡೆ, ಆ ಬನಾನಾ ಪೀಲ್ ಮೇಲೆ ಒಂದು ನೊಣಗಳು ಬರುತ್ತೆ ಚಿಕ್ಕದು, ಅದೇ ಫ್ರೂಟ್ ಫ್ಲೈ. ಓಕೆ, ಹೌಸ್ ಫ್ಲೈ ಚಿಕ್ಕದಿರುತ್ತೆ, ಫ್ರೂಟ್ ಫ್ಲೈ. ಯಾಕೆ ನಾವು ಮುಂಚೆ ಕಲ್ತ್ವಿ ನಾವು why Mendel took pea plants. Similarly, now we will also learn why did the T.H. Morgan took the Drosophila. ಯಾಕೆ Drosophila na? ಸರಿನಾ? They could be grown so simple on simple synthetic media in a laboratory. They complete their life cycle in about 2 weeks. Life cycle ಆಗೋಗುತ್ತೆ, and single mating could produce large number of progenies. Also, there was a clear differentiation of the sexes, male and female. ಈಸಿಯಾಗಿ ಡಿಫ್ರೆಂಟ್ ಕಾಣಿಸ್ತಾ ಇತ್ತು ಅಲ್ಲಿ, ಈಸಿಯಾಗಿ ಡಿಸ್ಟಿಂಗ್ವಿಷನ್ ಆಗ್ತಾ ಇತ್ತು. Okay, also many types of heredity variation that can be seen with the low power microscope can also be seen here. So ella ಕಾಣಿಸ್ತಾ ಇತ್ತು, everything was very visible here, and that was the reason why your T.H. Morgan took the fruit flies for his experiment. Now student, now I will teach you something called as linkage and recombination. Ready now? Students, ready now all of you? Tell me ready? Now I'll teach something called as your linkage and recombination. Okay, very simple, very easy to understand linkage and recombination. What is linkage? First I'll teach you. Okay, for example, let's say I have a friend here. Okay, I have a friend here. His name is Pandu. Okay, I have one more. Pandu has a friend called as Champa. Okay, Pandu has a friend called Champa. Now, between them, the bond is very close. If the bond between Pandu and Champa is very close, can I tell the linkage between them is very high? The linkage between them is very high. Now, if the linkage between them is very high, is there a chance that they will fight with each other? Not very well. So, if the linkage is very high, recombination is very low. Now students, Pandu has one more friend. Let's take Pandu has one more friend here. Okay, her name is, let's say, Sampa. Okay, now the bond between Pandu and Sampa, okay, is not very good. They're not able to, the bonding that is not there between Pandu and Champa. So can I tell between them linkage is very low? If there is no linkage between them, can I tell they'll fight with each other? Yes. So recombination is very high. Recombination between them is very high. Now what is the meaning of this linkage and recombination? Understand. I'll give you a perspective of a chromosome now. For example, this is a chromosome right here. Let's take, I have a chromosome right here. On this chromosome, I have one gene here, and I have one gene here. Let's say this is gene A, this is gene B, and this is gene C. Okay. Now these two genes are very close to each other. It means between them linkage is high, recombination is low. Gene A and C are very far. It means between them linkage is low, recombination is very high. This was a concept which we learned from your T.H. Morgan. That is, linkage is inversely proportional to recombination. Okay, recombination. ಇನ್ಕಮೆನ್ಸ್ ಪ್ರಪೋರ್ಷನಲ್. ಲಿಂಕೇಜ್ ಜಾಸ್ತಿ ಇದ್ರೆ ರೀಕಾಂಬಿನೇಷನ್ ಕಮ್ಮಿ ಇರುತ್ತೆ, ಸರಿನಾ? ಲಿಂಕೇಜ್ ಕಮ್ಮಿ ಇದ್ರೆ ನೋಡಿ, ಎ ಮತ್ತೆ ಸಿ ಮಧ್ಯದಲ್ಲಿ ಲಿಂಕೇಜ್ ಕಮ್ಮಿ ಇದೆ, ಸೊ ರೀಕಾಂಬಿನೇಷನ್ ವಿಲ್ ಬಿ ಹೈ. ಬಟ್ ವಾಟ್ ಇಸ್ ಲಿಂಕೇಜ್? ಮೀನಿಂಗ್ ಆಫ್ ಲಿಂಕೇಜ್. Linkage means physical association of two genes, gene A and gene B, on a chromosome. That is called as linkage. Linkage describes the physical association of genes on a chromosome, is called as linkage. One gene is here, one gene is here. Between them, linkage is very high. One gene is here, one gene is here. Between those two genes, not linked, recombination is high. Two very far, linkage is not there. Okay, then what is recombination? The term recombination was used to describe the generation of non-parental gene combination. ಯಾವ್ದು ಬೇರೆ ಬೇರೆ ಫಾರ್ ಇರುತ್ತಲ್ಲ, recombination ತುಂಬಾ ಹೈ ಇರುತ್ತೆ. ಸೊ ನಾನ್ recombinant parents ಬರುತ್ತೆ ಇಲ್ಲಿ. ಇಫ್ ಯು ರಿಮೆಂಬರ್ ಸ್ಟೂಡೆಂಟ್ಸ್, 9:3:3:1, ಇದು 9 ಮತ್ತೆ ಇದು 1, both of them were parental. Round and yellow, wrinkled and green, both are just like your parents ಯೂಸ್ ಮಾಡಿದ್ರಲ್ಲ ಡೈಹೈಬ್ರಿಡ್ ಅಲ್ಲಿ. ಬಟ್ ದಿಸ್ ಒನ್, these two are recombinant types, and recombination only happens when the two genes are very far from each other. Okay, so modern carried out several dihybrid cross experiments in Drosophila to study the genes that were sex linked. The cross was similar to dihybrid cross carried out by Mendel. Same marda. But what did he observe? He observed that, students, this was the insect he took. Okay, yellow bodied white eye female to brown bodied red eye male. Okay, he observed that, Morgan observed that two genes did not segregate independently. Independent segregation ಆಗ್ತಾನೆ ಇಲ್ಲ. ಆ 9:3:1 ratio ಬರ್ತಾನೆ ಇಲ್ಲ. ಯಾವಾಗ? When the two genes are very far from each other. There is no 9:3:1. So there is 9:3:1. If the two genes are very close, segregate ಆಗಲ್ಲ. ಉಲ್ಟಾ ಹೇಳಿದೆ, ಒಂದು ನಿಮಿಷ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. When the two genes are very close, if they are very close to each other, no segregation. Yake? If the two genes are very close to each other, can I say linkage is very high, recombination is low? When the two genes are very close to each other, linkage is high, recombination is low. ಅಕಸ್ಮಾತ್ recombination ಆಗಿಲ್ಲ ಅಂದ್ರೆ 9:3:1 ratio ಬರಲೇದಿಲ್ಲ. ಮೆಂಡಲ್ ವರ್ಕ್ ಮಾಡಿದ್ನಲ್ಲ, ಮೆಂಡಲ್ ವರ್ಕ್ ಮಾಡಿದ್ದು ಎಲ್ಲ ಜೀನ್ಸ್ ಒಂದಲ್ಲ, ದೇ ವರ್ ವೆರಿ ಫಾರ್ ಫ್ರಮ್ ಈಚ್ ಅದರ್ ಅಂಡ್ ಡಿಫ್ರೆಂಟ್ ಕ್ರೋಮೋಸೋಮ್ಸ್. ಅದಕ್ಕೆ ಅಲ್ಲಿ ಸೆಗ್ರಿಗೇಷನ್ ಆಗಿತ್ತು ಮೆಂಡಲ್ ಟೈಮಲ್ಲಿ, ಮೆಂಡಲ್ ವರ್ಕ್ ಅಲ್ಲಿ. ಬಟ್ ಇವನ್ ಮಾಡಿದಾಗ ಆ ಸೆಗ್ರಿಗೇಷನ್ ಬರಲೇ ಇಲ್ಲ, ಬಿಕಾಸ್ ದ ಟೂ ಜೀನ್ಸ್ ಆರ್ ವೆರಿ ಕ್ಲೋಸ್ ಟು ಈಚ್ ಅದರ್. ಸೊ ವಿ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ್ಯಾಟ್ ಹಿಯರ್, when the two genes are very close to each other on a chromosome, there is no segregation ಆಗಲ್ಲ. That is, recombination is very low, linkage is high, we are tightly bound. The segregation will only happen, the segregation or recombination will only happen when the linkage is low, when the two chromosomes are very far from each other. Okay, did not segregate independently of each other, and F2 ratio deviated from the 9:3:1 ratio. Okay, segregate agilla alli. So what has happened? Morgan and his group knew that genes were located on the X chromosome, and he quickly, when the two genes in a dihybrid clause were situated on the same chromosome, the proportion of parental gene combination were much more higher than the non-parental type. Artha eno, when the two chromosomes are present on the same chromosome, avaga parental combination high barutte. That is, parental combination high, recombination is low. Nodi, illi sick example. Illi nodi. Now here on this chromosomes, two genes are very close to each other. When this is happening, the parental, that is linkage, will be high barutte. Recombination will be very low. Why? The two genes are very close to each other. Recombination barnadilla illi. But here, this is you see, two genes are very far from each other. So linkage or parental combination is going to be very low. But recombination can be seen here. Recombination can be seen because the two genes are very far. Dura idre kai edko dura idre recombination is going to be high. Okay, recombination is going to be high. So with that, students, your linkage and recombination concept ನಿಮ್ಗೆ ಅರ್ಥ ಆಯ್ತು. See, two genes are very close to each other. Okay, close to each other. Recombination is very low. Recombination baralla. Two genes are very close to each other. Linkage is high, recombination is low. Recombination is low. Two genes are very far from each other. Different chromosomes ankoli. Same chromosome very far or different chromosome. In such case, recombination ನಿಮ್ಗೆ ಸಿಗುತ್ತೆ. Recombination ನಿಮ್ಗೆ ಸಿಗುತ್ತೆ. ಇಲ್ಲ recombination ಸಿಗಲ್ಲ. Okay. So linkage is high, recombination is very low. Linkage is high, recombination is low. Linkage is low, linkage ಕಮ್ಮಿ ಇದೆ ಇಲ್ಲಿ. Linkage ಕಮ್ಮಿ ಇದೆ, recombination ಬರ್ತಾ ಇದೆ. So ಈ ಕಾನ್ಸೆಪ್ಟ್ ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕಂದ್ರೆ this is the only formula which you know. This one right here. Linkage high, recombination low. Linkage low, recombination high. Tightly linked, loosely linked. Linkage high ಅಂದ್ರೆ ಏನು? ಇದು AB. Linkage high. AC is example of low linkage. Okay. That is called as your example of your TH Morgan. Okay. Crossing over ಆಗ್ತಾ ಇದೆ. Now students, ಈ scientist ಇದನ್ನಲ್ಲ TH Morgan ಒಂದು ಸ್ಟೂಡೆಂಟ್ ಇದ್ದ. That is called as Alfred Stewart Went. Alfred Stewart Went, he discovered something called as map units. That is how far and how close the two genes are present on a chromosome. ಈ ಜೀನು ಎಷ್ಟು ದೂರ ಇದೆ, ಈ ಜೀನು ಎಷ್ಟು ದೂರ ಇದೆ. These two genes are how far from each other. These two genes how close to each other. That is basically called as a map units. Alfred Stewart Went used the frequency of recombination between the gene pair on the same chromosome, measure of distance between the genes. ಎರಡು ಜೀನ್ ಮಧ್ಯೆ ಎಷ್ಟು ಡಿಸ್ಟೆನ್ಸ್ ಇದೆ. That is called as map units, also called as your Morgan unit, centimorgan, also called as your centimorgan or map units. Centimorgan or map units. Okay. Now students, now we will start something called as your polygenetic inheritance. Let's take exactly 20 seconds break. I'll drink some water and come. Okay, I'll drink some water and come. 20 seconds break, students. Let me drink some water. Okay, students, break done now. Now we'll start with something called as polygenetic inheritance. Okay. See, in the Mendelian genetics, we have been learning till now that there are two opposing contrasts. In Mendel, every single time he did his work, there was violet flower, white flower. The stem height was tall plant, dwarf plant. There was something called a seed shape, which was round or wrinkled. Nothing in the middle. But do you see that in your real life? If you go around right now, ಮನೆಯಿಂದ ಆಚೆ ಹೋಗಿ ನೋಡಿ, you will see human skin color to be in variety of them. That is, if you look at the human skin color, it is ranging from fair to dark and intermediate brown also. Human height is something not between, you'll never see something called as 6 feet and 4 feet. There is always intermediate also is present. That is 5'1", 5'5", 5'10", 5'11". All the intermediate height is also present. But according to Mendel, through contrasting traits is not possible. So there is something called as polygenetic inheritance. Polygenetic inheritance explains all of this intermediate different types of phenotypic expressions. See here. Such traits are generally controlled by 3 or more genes and are called as polygenetic traits. They are called as what? Polygenetic traits. Understand, very simple it is. When many genes control one trait is called as polygenetic inheritance. Many genes are there. For example, gene A, gene B, gene C. All the 3 genes are responsible for the human skin color. That is going to be your polygenetic traits. And let me tell you, students, these polygenetic traits, right? They are controlled by environment also. You go stand outside in the sun, tan bartira. So polygenetic traits are controlled by many genes. But apart from all that, students, polygenetic traits are said to be additive in nature. They are additive. Now, what is the meaning of additive? The meaning of additive here is addition. Plus agate illi. For example, let's take I have gene A, gene B, and gene C. I have 3 genes here, 2 alleles for all of them. 2 alleles. Okay. Gene A, gene B, gene C. Okay. Similarly, I have, like, let's take gene A, gene B, gene C. I have one more gene A, gene B, gene C. Let's take, I have one more gene A, gene B, gene C, gene C. Let's take, I have one more small a, small b, small b, small c, small c. Now students, do you see here? In the first example, we have all dominant alleles. All dominant alleles are present. So the skin color is going to be completely dark. Here, all recessive alleles, skin color is going to be the lightest. It's going to be the lightest skin color it is going to be. Now, do we have intermediate in the middle? Yes, we have intermediate. Here we have somewhat, let's say here we have somewhat brown. This is much more lighter in nature. Lighter in nature. So, can I say that more number of dominant alleles, darker is the effect. Darker or the stronger is the effect. Here we have dark skin color. Less number of dominant alleles, very weak is the effect. So, stronger the effect means more dominant alleles. So, this is called as additive effect. Additive effect. So, additive effect explains you that the more number of dominant alleles, more stronger is the effect. Okay, you can see here also. All dominant alleles, darker the effect. Lighter alleles, lighter is the effect. That is called as your additive effect, which is observed in the polygenetic inheritance. So first point is what, students? First point is many genes controlling one trait. Can you see here? Gene A, gene B, gene C. All of these genes are controlling one trait, that is skin color. That is skin color. All of them are controlling skin color. Many genes, one trait. It is additive in nature, and also it is affected by environment. Affected by the environment. Okay. That is the polygenetic inheritance. Okay. See, one more example is given here. Skin color. Okay. As expected, genotype with 3 dominant alleles and 3 recessive alleles will have intermediate skin color. So 3 dominant, 3 recessive, intermediate in the middle. Okay. In this manner, the number of each allele in the genotype would determine the darkness or lightness of the skin color. So more number of dominant, darker is the skin color. Okay, very dark. Now students, that was your polygenetic inheritance. Many genes, one effect. One effect. Now next concept is called as pleiotropy. Very important, many questions have come from here. Now pleiotropy is the opposite of it. Opposite of polygenetic. In pleiotropy, what happens is, one gene, many effects. So can you see here, a single gene, students, a single gene will have multiple expressions. Multiple expression. ನಿಮ್ಮ ಮಮ್ಮಿ ತರ ಇದು ಅಮ್ಮ ಎಲ್ಲ ಕೆಲಸ ಮಾಡ್ತಾ ಇದ್ರೆ ಮನೆಯಲ್ಲಿ cook, clean, take care of you, everything. This is a mummy gene, okay, housekeeping gene. So single gene will have multiple phenotypic expressions. Polygenetically many genes, one effect. Gene A, gene B, gene C, skin color. In pleiotropy, a single gene, multiple effects. Okay, such an expression is called as multi-phenotypic expression. That is called as pleiotropy. That is called as what? Pleiotropy. Okay. The underlying mechanism of pleiotropy is in the most cases, is the effect of gene on metabolic pathway which contributes towards a different phenotype. So how does it work? Pleiotropic gene affects the metabolic pathway. Remember that. An example of this disease is phenylketonuria, students. Understand, very simple example they've given you. PKU disease, phenylketonuria disease. Now, phenylketonuria disease, what happens? Phenylalanine is not converted to tyrosine. Phenylalanine is not converted to tyrosine because there is one enzyme is missing. The enzyme is called as your phenylalanine hydrolase. If this enzyme is missing, then we get a disease called as phenylketonuria disease, PKU disease. PKU disease. Yav barati PKU disease? When this enzyme is completely missing. Enzyme is missing. Okay. Now, when this enzyme is missing, and because of this mutation, because of this, what do I say? One defect in the gene. One defect in one gene can have multiple phenotypic expressions. That is mental retardedness, reduction in hair, skin pigmentation. Can you see? Multiple phenotypic expression because of effect in one gene. So one gene, multiple phenotypic expressions. Now students, that was polygenetic inheritance and pleiotropy. Now we come to something called as sex determination. Sex determination is a mechanism which helps us to identify the sex of the offspring. Is it male or is it female? Okay. But before we go to sex determination, let's understand a small basics behind it, or a little bit of history. Who was the scientist involved? For example, if I say pea plant, you will think of Mendel. If I say Drosophila, you will think of T.H. Morgan. If I talk about evolution, you will think about Charles Darwin. But when I say sex determination, tell me which scientist comes to your mind? Probably no one, because you didn't study this. Okay, so let me tell you, the scientist which is supposed to come to your mind is called as Henking. Henking is a scientist whose main contribution was towards sex determination. See here, Henking. The mechanism of sex determination has always been puzzled before the genetics of genetists. Okay, the initial clues about genetic or chromosomal mechanism of sex determination can be traced back to some experiments carried out in the insects. So starting ಅಲ್ಲಿ ವರ್ಕ್ ಮಾಡಿದೆಲ್ಲ ಇನ್ಸೆಕ್ಟ್ಸ್ ಮೇಲಿತ್ತು, ಎಲ್ಲ ವರ್ಕ್ ಕೂಡ ಇನ್ಸೆಕ್ಟ್ಸ್ ಮೇಲಿತ್ತು ಮುಂಚೆ, ಸರಿನಾ? Now understand students. Now Henking could trace a specific nuclear structure in all through spermatogenesis in his few insects. He observed the spermatogenesis of few insects and he observed that in 50%, this is X, this is Y, in 50% of the sperm, there was an X structure was present. In 50% sperm, the X structure was absent. So Henking observed that during spermatogenesis, 50% of the sperm has the X structure, that is the X chromosome, later we found out, and 50% did not have it. Okay, so it was observed by him that 50% of the sperms received this structure after spermatogenesis, whereas the 50% of sperm did not receive it at all. Okay, and later he called it as X body because yavattu kuda gottalla biology maths alli, if you don't know something, call it as X. Gottilla aste X body. Okay, so he called it as X body but could not explain its significance. Further investigation by other scientists led to the conclusion that the X body was in fact the X chromosome. So Henking was a scientist whose main pioneer work was on the sex determination. Now students, I'll teach you there are two main concepts here called as male heterogamety and female heterogamety. What is male heterogamety? Understand first of all. Male heterogamety. Male heterogamety is a condition in which the males decide the sex of the offspring. Example, humans only. Humans, female is, in humans, the female is XX, male is always XY. So the male will determine the sex of the offspring. For example, if X chromosome from here, X chromosome here meet, we get XX, that is a female child. If this X, same X and this Y meets, we get a male child. So here the male determines the sex of the offspring. That is called as male heterogamety. Hetero means different, gamete means gametes. So male has two different gametes here. The same goes for grasshopper also. Can you see here? The males have XO and the females have XX. Again, the male determines the sex of the offspring. Now, what about in the case of female heterogamety? In the case of female heterogamety, the females will decide the offspring sex. See here, this is the female ZW. In the case of birds, ZZ is going to be the male. ZZ is going to be the male here. It's going to be the male. Okay, it's going to be the male. Same goes for butterfly. ZO is going to be the female, different type of gametes. ZZ, which is a common thing, which is going to be the male. So in the case of male heterogamety, the males decide the sex of the offspring. In the case of female heterogamety, it is the females which decide the sex of the offspring. Okay, it is going to be male or female. It is written by here. Female determines here because different type of gametes. Here, male has different type of gametes here. That is called as male heterogamety and female heterogamety. Examples tumba importantly in examination, in KCET examination, there are many questions based on this concept here. Many questions are there on this concept. Okay. Now, apart from all that, students, there's one more concept here. There's one more concept given your NCERT, that is called as haplodiploidy. Ready, all of you students? Ready? Haplodiploidy. Now, haplodiploidy is mainly observed in the case of honeybee. So what exactly happens in haplodiploidy? Now here understand, students, the sex of the offspring mainly is determined by the set of chromosome. Yes, chromosome ideyala, other male, it is going to be male or female is determined. For example, let's take here, usually males are haploid here. Males are completely haploid here, while the females are diploid. Here the 2n is equal to 32. 2n is equal to 32 chromosomes. Here 16 chromosomes, right? 16, 16. Here 16 chromosomes. 16 chromosomes. Okay. So males are haploid, females are diploid here always. Okay. So this male is producing a gamete. Let's take a sperm. Here the female is producing a gamete, that is the egg. Okay. Now this most important thing here is this egg and this sperm both are going to be haploid in nature. Haploid in nature. Now here we have 2n is 32, 2n, 2n. Tell me, should we have mitosis here or should we have meiosis here? Obviously, change in the ploidy means meiosis. Meiosis. Here, ploidy remains the same, nn, so mitosis. This is what is very important to understand. So in the case of your sperm production, it is with the help of your mitosis. In the case of your egg production, it is with the help of meiosis. Now finally we have the gametes here, sperm and egg. Let's fuse them now. Let's fuse them. Okay. If you fuse sperm plus egg, we end up getting the female child. The zygote will develop into female. Why? Because n plus n is 2n and the females are always 2n. So males are never born like fertilization. Males are never born out of fertilization here. Never. Not possible only. Okay. So this is a female here. 2n. Now this female right here, students, can be a worker or a queen. It can be a queen bee, or it can be a worker bee. Queen bee or it can be a worker bee. Now you'll ask me, sir, where are the males then? Understand. Now this egg right here, right? This egg right here develops directly into the male. This here directly develops into the male. And haploid. And this process where the males are formed directly from the egg to the male, such a process is called as parthenogenesis. It's called as parthenogenesis. Parthenogenesis right here. That's how we end up getting the male. Male here, female as a result of fertilization. Diploid, haploid males. Now there is a small concept here, understand students, very interesting and small concept here. That is the males here, students. Now this male right here, which is born here, does it have a father? Tell me, does it have a father now? No, no father here. So no father. Now can this male right here, eventually this male will produce the sperm and undergoes fertilization. The sperm undergoes fertilization and get the female only. So can I tell this male can never have a son also? No father, no son. Not possible, because the male will always produce a female. There is no son. But does this male, which is born from a female, see, for example, this female will eventually give a son. Will give a son by parthenogenesis. Now this son right here, same male or the son here, doesn't have a father, but it has a grandfather. Okay, has a grandfather. You can have a grandfather. Not really. E male gave father antu illa. But if this female has to be born, it has to be born from a male only. So it can have a grandfather. Now tell me, students, this male is producing a female. This female is producing a son. So can I tell there will be a grandson? Yes, there will be a grandson. So in the case of your haplodiploidy, there is no father, there is no son, there is a grandson and grandfather. Amazing concept, no? Amazing. That is your haplodiploidy. Sex determination. Okay. We have seen this already. Grasshopper, XO. Male heterogamety. Okay. Males are XO, females are XX. Males determine the sex of the offspring. Okay. Offspring. Same goes for humans also. Drosophila. In humans and Drosophila, both are male heterogamety. Males have one X and Y chromosome, while the female have XX chromosome. Females are X chromosome. So Drosophila and humans are the examples of male heterogamety. Same thing in birds. We have Female heterogamety, ZW, female. Males are ZZ. Okay. Now, what about sex determination in the case of humans? Sex determination in the case of humans is male heterogamety, and usually the male decides the sex of the offspring. Males has XX, XY, females have XX. Okay. And remember, students, ನೀವು ನೋಡಿರಬಹುದು ಮೂವಿಲೆಲ್ಲ they always blame the female for not giving a male child, but it is the males who decide, not the female. Female only has one chromosome, that is X chromosome. It is the males who decide the sex of the offspring. Okay. Now, students, next we come to the concept of mutation. Now, what is mutation? All of you are aware. Cancer barattala, cancer. So mutation is basically sudden inheritable change in the chromosome. Into genes ali in a small change aito, but you can inherit it, and it is going to be there throughout a generation now. That is called as mutation. Okay. Mutation is a phenomenon where results in alteration of DNA. DNA ಸ್ವಲ್ಪ change ಆಗುತ್ತೆ ನಾವು. Okay. And subsequently results in a change in the genotype and phenotype of an organism. Obviously, DNA ಅಲ್ಲಿ ಏನಾದರೂ change ಆದರೆ phenotype ಮತ್ತು genotype change ಆಗೇ ಆಗುತ್ತೆ for sure. ಹಂಗೆ ಇರಕ್ಕಾಗಲ್ಲ. ಸರಿನಾ? In addition to recombination, mutation, other phenomena that leads to variation. So, genetic variation ಗೆ ಎರಡು main ಕಾರಣ ಇದೆ. Recombination ಆಗ್ಬೋದು during meiosis, and mutation can happen. That is, change in the genetic makeup. Okay, change in the genetic makeup. Now, what exactly is mutation now? What exactly is mutation? In mutation, there is a change in the DNA. There is a change in DNA, or there is a change in the gene also. Gene is a part of DNA, all of you know. Gene is the smallest segment of DNA. Gene is part of DNA. Gene is part of chromosome. So any change in chromosome, there will be change in gene also. Okay. So there can be loss or gain of or insertion or duplication of segment of DNA. For example, if this is a normal set of chromosomes, there can be a change in the chromosome. Yes. Change in the chromosome. There can be completely insertion or deletion of your nucleotide bases. For example, here let's take, we have TCGG, or let's say we have GAC. Completely remove it. That is called as deletion. Insertion. So when we insert a nitrogenous insert a base pair and remove a base pair, there will be mutation. Now there are two main examples here. One is called as your point mutation, where a single nucleotide is removed. Can you see here? Mutation arises due to change a single base pair of DNA. For example, here we have TAACT. We will have TAACC. A single base pair has been changed. That is called as point mutation. Okay. Other one is called as frame shift mutation. See here. This is the original sequence. Original sequence is here. Original sequence, let's say, read this one. We have TAACTGCAAGC. Now what happens is, what if there is a removal of one base pair? Let's take, I removed T, I removed. Now if I'm not adding anything, what will happen? If I don't add anything, the entire frame will move forward. This entire frame was moving one forward. So because this one entire frame was moving, initially it was TAACT. Now it is becoming TAACC. Why? Because this entire frame is moving forward. It is moving forward. That is called as your frame shift mutation. Okay. Now, for example, this is moving this side. Now, what if I add something? Then it will move this side. So if I remove front, if I add something, moving backwards. Okay. That is called as your frame shift mutation. The entire frame is moving forward or backward because of addition or removal of something, not replacing. Okay. That is called as frame shift mutation and point mutation. Now you might ask me, sir, how are this mutation being caused? Is there any reason for it? Yes, there is. Mutagens. They're called as mutagens. It could be harmful gamma rays, UV rays. All of this can cause the cancer in the body. Okay. Many chemicals and physical factors, mutation, mutagens, UV radiation, UVB, UVC, UVA. All of these are mainly causing the mutation. Okay. So students, remember, mutation can cause a change in the entire DNA. The next concept is called as your pedigree analysis. Very simple, very easy. In dog biscuit alaidu, pedigree is basically let's say it's a map which tells you a history about your family. ಒಂದು ಇಂಡಿಯಾ ಮ್ಯಾಪ್ ಅನ್ಕೊಳ್ಳಿ, ಮ್ಯಾಪ್ ಇದು. ಈ ಮ್ಯಾಪ್ ಅಲ್ಲಿ ನಿಮ್ದು ಎಂಟೈರ್ ಫ್ಯಾಮಿಲಿದು ಹಿಸ್ಟ್ರಿ ಇರುತ್ತೆ, ಯಾವ ಪರ್ಸನ್ಗೆ ಆ ಡಿಸೀಸ್ ಇದೆ ಎಲ್ಲ ಹಿಸ್ಟ್ರಿ ಇರುತ್ತೆ. ಅಂಡ್ ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್ we can identify any diseases there in the family and cure it also. See here, for example, this is the entire map of your generation, parents, children, grandchildren, great grandchildren, full family history irutte, and we can find if anyone is deceased. In pedigree analysis, inheritance of a particular trait is represented in a family tree. For example, let's say ಇಲ್ಲಿ ಈ ಪರ್ಸನ್ಗೆ ಸರಿನಾ, ಈ ಹ್ಯೂಮನ್ಗೆ ಏನೋ ಒಂದು ಡಿಸೀಸ್ ಇದೆ, ಅದನ್ನ ರೆಡ್ ಕಲರ್ ಮಾರ್ಕ್ ಮಾಡಿ, red color mark ಮಾಡಿ, red color mark ಮಾಡಿ. So every single pedigree students, every single pedigree has certain symbols. For example, square ಅಂದ್ರೆ ಮೇಲು, round ಅಂದ್ರೆ ಫೀಮೇಲು, red color ಅಂದ್ರೆ affected. Okay, mating ಅಂದ್ರೆ single line, mating ಅಂದ್ರೆ single line, mating between relatives, that is consanguineous mating ಅಂದ್ರೆ ಎರಡು ಲೈನ್ ಇರುತ್ತೆ, ಎರಡು ಲೈನ್ ಇರುತ್ತೆ. Okay, parents above the children below ಯಾವತ್ತು ಸರಿನಾ ಈ ತರ ಇರುತ್ತೆ. Okay, parents with male child affected, ನೋಡಿ ಇಲ್ಲಿ ಮೇಲ್ ಚೈಲ್ಡ್ affected ಆಗಿದೆ, ಮೇಲ್ ಚೈಲ್ಡ್ affected ಆಗಿದೆ. This is called as your entire symbols. ನಿಮ್ಮ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ they can ask you directly these symbols without even thinking. They'll ask you which is a symbol for male, which is a symbol for consanguineous mating. ಆ ತರ ಕ್ವಶನ್ ಡೈರೆಕ್ಟ್ ಆಗಿ ಬರುತ್ತೆ. ಅದು ಬಿಟ್ರೆ they have two different examples given your NCERT, two different examples they have given. ಇದೆಲ್ಲ ಬಿಟ್ಟರು ಕೂಡ ನಿಮಗೆ ಕ್ವಶನ್ ಕೇಳಬಹುದು, what is autosomal dominant disease, autosomal recessive disease example ಕೊಡಬಹುದು. But 99% of time they will not give you this. The only thing which they will ask you is right now is two different examples which is given in your NCERT itself. One is autosomal dominant, one is recessive. See here, the example for your autosomal dominant is myotonic dystrophy. Idu myotonic dystrophy, this is your autosomal dominant. For example, myotonic dystrophy, this one, myotonic dystrophy. Okay, it's a dominant trait, affected, it is never skipping a generation skip ಆಗ್ತಾ ಇಲ್ಲ. Affected parents can have unaffected children. Now one is autosomal recessive, that is example is sickle cell anemia, it can skip generation, recessive in nature, and upon that unaffected parents can have affected children. Okay, so that is the small traits of your pedigree analysis. And let me know in the comment section if you want a small video of pedigree analysis, one small 10 minutes video to identify pedigree analysis which will help you in preparation. Okay, let me know in the comment section if you want it. Okay, now students, after pedigree we come to the last topic of your syllabus. Last topic idu ee chapter do, that is basically the different type of diseases here. For example, we have diseases can be broadly classified into two separate categories here. One is called as Mendelian disorder and other one is called as your chromosomal disorder. Okay, well, what is the difference here? Understand, Mendelian disorder is basically change in a single base pair or single gene mutation, single gene, while chromosomal disorder is change in the chromosome number itself. Chromosome number change agogutte in chromosomal disorder. Okay, so Mendel disorder determined by alteration of mutation in the single gene. Okay, single gene change ಆಗ್ತಾ ಇರುತ್ತೆ. These disorders are transmitted to the offsprings on the same lines as we have studied in the principles of inheritance. So Mendelian disorders follow the Mendelian principles. Other one is chromosomal disorders. Okay, chromosomal disorders are caused due to absence or excess of abnormal arrangement of one or more chromosome. Now students, I will show you an entire table. Just look at this table and remember this table because many questions will come directly from this table right here. Ready students? Okay, one second, where is the table? Right here. Can you see? This is a table which I have made for you for genetic disorders with examples every single place. Mendelian disorder, alteration in a single gene. Chromosomal disorder, genetic mutation or chromosomal aberration. The meaning of chromosomal aberration is change in the chromosome number. Chromosome number change agutte. Okay, now Mendelian we have autosomal dominant, autosomal sex linked, autosomal dominant recessive, sex linked. Examples. The only example of autosomal dominant given in your syllabus is myotonic dystrophy. Autosomal recessive, PKU, phenylketonuria, sickle cell anemia, thalassemia. Three different examples. Example of sex linked, color blindness, hemophilia. Then we have chromosomal disorders, autosomal Down syndrome, sex-linked Turner syndrome, and Klinefelter syndrome. ಈ complete table ನಿಮಗೆ ನೆನಪಿರಬೇಕು. If I ask you, tell me the example of autosomal recessive. ಕಣ್ ಕ್ಲೋಸ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಟೇಬಲ್ ನೆನಪು ಮಾಡ್ಕೊಳ್ಳಿ. ರೈಟ್ ಹ್ಯಾಂಡ್ ಸೈಡ್ PKU, sickle cell anemia, thalassemia. Clear all of you? So ಈ ತರ ನಿಮಗೆ ಈ ಪೂರ್ತಿ ಟೇಬಲ್ ನಿಮಗೆ ನೆನಪಿರಬೇಕಿದು. Entire concept of your genetic disorders. Okay, genetic disorders. Beautiful table is here. Okay. So we have autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive. All of them are present in Mendelian disorder. Mendelian disorder. All of these Mendelian disorder students, all of these Mendelian disorders can be studied with the help of pedigree also. And remember, in pedigree, we always study Mendelian disorder only. Nothing extra. Nothing extra. Disorder. Okay. Now students, let's get to it. One by one now. One by one, can we start? All of you drink some water and we'll start. All of you drink some water and we'll start with the first Mendelian disorder that is called as color blindness. Okay, color blindness. Drink some water, all of you. Drink some water. Now, let's start with something called as color blindness. Color blindness is a disorder, students, in which a human cannot distinguish between a red and green. ಅವರು ಡ್ರೈವ್ ಮಾಡ್ತಿದ್ರೆ ಯಾವ್ದು ರೆಡ್ ಕಲರ್ ಯಾವ್ದು ಗ್ರೀನ್ ಕಲರ್ ಸಿಗ್ನಲ್ ಗೊತ್ತೇ ಆಗಲ್ಲ ಅವ್ರಿಗೆ, ಸರಿನಾ? ಸೊ ದೇ ಕಾಂಟ್ ಡಿಫರೆನ್ಶಿಯೇಟ್ ಬಿಟ್ವೀನ್ ರೆಡ್ ಅಂಡ್ ಗ್ರೀನ್ ಹಿಯರ್. ಓಕೆ, those are called as, that is called as color blindness. Color blindness. Okay, now this is a color blindness. Students, first of all tell me, color blindness ಯಾವ್ದು ಡಿಸಾರ್ಡರ್ ಇದು? If you go here, color blindness ಇಲ್ಲಿ ಬರ್ತಿದೆ, color blindness ಇಲ್ಲಿ. So it's a sex-linked recessive disorder. Sex-linked recessive disorder. Okay, it's a sex-linked recessive disorder. Sex-linked recessive disorder due to defection in the red and green cones of eye, resulting in the failure to discriminate between red and green color. So ಅವ್ರಿಗೆ red color ಯಾವುದು, green color ಯಾವುದು ಗೊತ್ತೇ ಆಗಲ್ಲ ಅವ್ರಿಗೆ. Okay, now it may be on it occurs only in 8% of males and 0.4% females. So the females is ತುಂಬಾ rare phenomena, and most of the males are affected in the world because of color blindness. Color blindness. So males have only one X chromosome and the female have XX. Since the X recessive, so males are very affected. Males are affected here. Okay. Now the son of a woman who carries the gene has 50% chance of being color blindness. Okay. That is, for example, I have female here. The son of a woman who carries the gene has 50% chance of being colorblind. Okay. Now, for example, let's say a female. The female is XX, and let's say the female is carrying a genetic disorder. She's a carrier. Female is a carrier. The son here can get this, will get the X chromosome from here, or it can get X chromosome this one. So there's a 50% chance that the son can be colorblind. Okay. The mother is not herself colorblind because the gene is recessive. So mother, this person is not colorblind. But if the male is something like this, the male is going to be colorblind, but the female is going to be carrier. So most of the time, the females are carrier and males are generally affected because we only have one X chromosome. Akasmad, E chromosome bambitre, they are colorblind for sure. Okay, that means that the effect is suppressed by her matching dominant normal gene here. So usually the males are generally affected here and the females are generally carrier. One example ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ, unaffected father. Okay, so let's take unaffected father XY, carrier female. Okay, carrier female. Okay, now what are the gametes we get here? X and Y. Gametes here XC and X. Let's draw Punnett square, all of you. Male female. Let's do male XY, female is XC, carrier. Now what do we get here? This is a carrier female, colorblind male, a normal female, and a normal male. So whenever there is a mother, whenever a mother is carrier, there is a 50% chance. Can you see? 50% chance that the son is going to be colorblind. 50% normal. So this is called as the colorblind. Now I'll give you one more example. Let's take one more example. The male is, let's say, colorblind now. Male is colorblind and the female is carrier now. Female is carrier, the male is colorblind. Colorblind man marries a carrier female. Now tell me, what are the possibilities now? X, C, gametes, X, C, the gametes. Do the Punnett square, X, C, Y, X, C, and X. When this is the case, one colorblind female, colorblind male, carrier female, and a normal male. So in examination they can ask you, tell me, what is the percentage of females being colorblind here? Female is 100%. 50% chance a female is going to be colorblind. Okay. Now I'll ask you one more question. What is the percentage of males being colorblind? 50% are going to be colorblind. This is 100% totally. 50% is going to be colorblind. Now what if I ask you, what is the percentage of offspring being carrier here? Offspring being carrier. Now when I say offspring, you have to take on entire thing, males and females both. This is going to be normal. This is going to be colorblind. So in such cases, 25%, 100%. 25% is going to be carrier. Okay. So when I say, remember the question, they're asking you male, female, or the offspring. Male, female, and if take only male, only female. But when they say offspring, entire all four should take. So 100% avaga. So avaga 25 agutte. Illa andre bari male, female andre 50-50 agutte. 50-50 agutte. Okay. That is about your color blindness. A daughter will not be colorblind unless her mother is a carrier and father is colorblind. Nodadvalla. Mother is a carrier, father is colorblind. Ee case matra a female can be colorblind. Okay. So students, many questions tumba barate questions colorblind inda. Especially colorblind inda many questions. So practice ಮಾಡಿ ಇದನ್ನ ಸರಿನಾ. ಏನು ಡೌಟ್ ಇದೆ ನನಗೆ ಕೇಳಿ, ಕಮೆಂಟ್ ಅಲ್ಲಿ ತಿಳಿಸಿ ನನಗೆ ಸರಿನಾ. ದಟ್ ಇಸ್ ಯೋರ್ ಕಲರ್ ಬ್ಲೈಂಡ್ನೆಸ್. ನೌ ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಹಿಮೋಫಿಲಿಯಾ. ಓ ಇದಕ್ಕೆ ಬಂದಿದೆ, ಇದು ಅದು ಮುಂದೆ ಇರಬೇಕಿತ್ತು. ಹಿಮೋಫಿಲಿಯಾ. ಓಕೆ, ಹಿಮೋಫಿಲಿಯಾ ಒಂಥರ ತುಂಬಾ ಡೇಂಜರಸ್ ಡಿಸೀಸು. ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ಹಿಮೋಫಿಲಿಯಾ ಒಂದು ಸ್ಮಾಲ್ ಕಟ್ ಆದ್ರೆ ಅನ್ಕೊಳ್ಳಿ ನಿಮ್ಮ ಬಾಡಿ ಮೇಲೆ, ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಬ್ಲೀಡ್ ಆಗ್ತಾನೆ ಇರುತ್ತೆ, ಸ್ಟಾಪ್ ಮಾಡೋಕ್ಕೆ ಆಗಲ್ಲ. That is called as bleeder's disease. Okay. Hemophilia is sex-linked recessive disorder. Sex-linked recessive disease. Sex-linked recessive. So ನಿಮ್ಮ ಇನ್ಸೈಡ್ ಇರೋದು ಎರಡು ಎಲ್ಲ ರಿಸೆಸಿವ್. ಸರಿನಾ, ರಿಸೆಸಿವ್ ಇರೋದು ಎಕ್ಸಾಂಪಲ್ಸ್ ಕೂಡ. Sex-linked recessive disease. ಕಲರ್ ಬ್ಲೈಂಡ್ನೆಸ್ ಆಯ್ತು, ಹಿಮೋಫಿಲಿಯಾ ಈಗ bleeder's disease. Okay, bleeder's disease. Okay. Now, cell recessive, in which a single protein that is part of a cascade of protein. Cascade ಅಂದ್ರೆ ಲಾರ್ಜ್ ನಂಬರ್ ಆಫ್ ಪ್ರೋಟೀನ್ಸ್, ಒಂದು ಸ್ಮಾಲ್ ಪ್ರೋಟೀನ್ ಇರುತ್ತೆ, ಆ ಪ್ರೋಟೀನ್ ಅಲ್ಲಿ ಡಿಫೆಕ್ಟ್ ಮಾಡ್ಬಿಡುತ್ತೆ ಸರಿನಾ. ಇನ್ವಾಲ್ವ್ ಇನ್ ಕ್ಲಾಟಿಂಗ್ ಆಫ್ ಬ್ಲಡ್ ಇಸ್ ಎಫೆಕ್ಟೆಡ್. ಡ್ಯೂ ಟು ದಿಸ್ affected individual, a simple cut would result in non-stop bleeding. ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಬ್ಲೀಡ್ ಆಗ್ತಾನೆ ಇರುತ್ತೆ, ಸ್ಟಾಪೇ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ as bleeder's disease, also called as hemophilia. ಓಕೆ, ಅಗೇನ್ ಇಲ್ಲೂ ಕೂಡ ಫೀಮೇಲ್ ಎಫೆಕ್ಟ್ ಆಗೋದು ತುಂಬಾ ರೇರ್. ವೈ? X-linked recessive it is. X-link. So female ಗೆ ತುಂಬಾ ರೇರ್ ಆಗುತ್ತೆ. Males are generally more affected here again. Right? Heterozygous female carrier for hemophilia may transmit disease to her son. For example, here we have a female, carrier female. ನೋಡಿ, ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ, ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Let's take male is XY, no disease. Female is hemophilic in nature. Now, when we do the gametes and do the Punnett square, we have XY, XH, and X. Female is going to be carrier. This male is going to be hemophilic. Okay? Female was carrier, but the son is already hemophilic in nature. So again, there is a 50% chance that the carrier female, normal male, will give a hemophilic son. So sons will have generally affected here, generally more affected here. Okay. Males are more affected here. The possibility of female becoming hemophilic extremely rare, because the mother of such female has to be at least carrier and the father has to be hemophilic. ಏನಪ್ಪ ಇದು ಹಂಗಂದ್ರೆ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. If the female has to be female has to be hemophilic, then what is the possibility? Female has to be a carrier. Okay, female has to be a carrier. Female has to be a carrier. The female has to be a carrier and the father has to be hemophilic. Father has to be hemophilic. Hemophilic. Gametes madadmele ಏನು ಬರುತ್ತೆ ಇಲ್ಲಿ? Male is XHY, female is XH and X. Hemophilic gene. Can you see? The female is becoming hemophilic here. And also the male is becoming hemophilic. Carrier female and normal male. Same concept we also applied for color blindness also. Same concept is also here. That is, for a female to become color blind or hemophilic, it is very rare. Males are generally more affected here. Okay. See here. And the family pedigree of Queen Victoria. Our family the disease is there. Our family the disease is there, where males are generally more affected here. Okay. Now next one is called as sickle cell anemia. Now X-linked mugito. Sickle cell anemia was part of autosomal recessive. ನೆನಪಿದೆಯಾ ನಿಮ್ಮೆಲ್ಲರಿಗೂ? Sickle cell anemia. Can you see? CKU. Autosomal recessive sickle cell anemia. SCA. Sickle cell anemia was here. Okay. Sickle cell anemia. ಏನಾಗುತ್ತಪ್ಪ sickle cell anemia ಮತ್ತೆ mutation ಕೂಡ. It is an autosomal linked recessive disease. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತದು. Trait that transmit from parent to the offspring when both parents are carrier for the gene. Both of them are carrier, then it is completely transferred. I'll show you. See, the disease is controlled by a single gene or pair, that is HBA and HBS. HBA is normal, HBS is sickle cell. For example, students, see this. If a particular parent has HBA and HBA, or any human has HBA, it is normal condition. Okay. HBS and HBA, this person is going to be carrier. Okay. HBS is here. HBS and HBS, this person is going to be affected, completely affected, because both the alleles are present which are sickle cell in nature, and this person will have sickle cell anemia. Okay. Now heterozygous condition will transmit from parent to the offspring. Heterozygous condition, this person can transfer it to the offspring easily. Offspring kalaspodinna. The disease is controlled by a single pair of alleles, that is HBA and HBS. HBA unaffected. HBA, HBS, both of them are carriers. HBS is affected. It is affected. So the example given you here is, one is a carrier father and carrier mother. Let's see, we also do the cross. Let's do the cross ourselves. Carrier male and carrier female. Okay, let's take HBS, HBA. This is a carrier male. HBS and HBA. This is carrier female. Let's do the table. Here we'll get HBA and HBS from here. Here we will have HBS and HBA. Now tell me, students, this person is going to be carrier, HBA, HBS, carrier. This one. Next one is going to be HBS and HBS. This person is going to be affected. HBA and HBA. This person is going to be completely normal. HBA and HBS. Yak HB? Hemoglobin entirely. This person is going to be normal. This person is going to be sickle cell anemic. This person is going to be one second. This carrier. HBS is this person is going to be normal. both are HbA. This person is again going to be a carrier. That is the beauty of sickle cell anemia. Okay, when both are present, HbA, HbS, is going to be sickle cell anemic. Now understand what exactly happens in sickle cell anemia. Out of the three possibilities genotype, only homozygous individuals of HbS, HbS show disease phenotype. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತು, HbS ಮಾತ್ರ ಇದ್ರೆ ಮಾತ್ರ that person is sickle cell anemic in nature. Now what is going to happen if the person is sickle cell anemic? ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ ಏನಾಗುತ್ತೆ ಅಂತ, ಸರಿನಾ? The defect is caused by substitution of glutamic acid by valine. You know, this right here is the all of you can see this now? Yes, all of you can see this. This is normal hemoglobin. Here mRNA has HGAG. GAG codes for glutamic acid and the sixth position of a beta globin chain. This is called as HbA protein, HbA peptide. Now because of the mutation, because of the point mutation in the mRNA, we have, instead of GAG, we have GUG here. Why? That is because of change in the normal gene also. Here we have GAG, GTC it became here. So CAC, GUG is here. Here we have GAG. So there's a change in the gene itself. One point mutation. What is happening here? Point mutation is happening. Because of point mutation, adenine is replaced by thymine. And because of that we have glutamic acid here. And because of one point mutation, we have valine here. So glutamic acid is changed to valine at the sixth position in the beta globin chain. This is valine now. Now such a change will cause a complete change in the structure of the hemoglobin. Because of this, defect is caused by substitution of glutamic acid by valine at the sixth position in the beta globin chain of hemoglobin. That is causing the change in the complete shape of hemoglobin. Can you see? Your RBC was biconcave. It became sickle shape. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing a change in the shape of the RBC from biconcave to sickle cell anemic. And because of this condition, the particular person will have very less oxygen and they'll become anemic in nature. Sickle cell anemia, anemic in nature. Okay, that was your SCA. Now we come to one more disease called as PKU. PKU again is a type of autosomal recessive trait. Here, ನೆನಪಿಲ್ಲಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇದು ಈ ಪಾರ್ಟ್ ಮಾತ್ರ ಮರೆಯಕ್ಕೆ ಹೋಗ್ಬೇಡಿ ನೀವು. ಈ ಪಾರ್ಟ್ ಮಾತ್ರ ಮರೆಯಕ್ಕಂತ ಹೋಗ್ಲೇಬೇಡಿ. ಇದು ನೋಡಿ, PKU ಥಾಲಸೆಮಿಯಾ ಬೋತ್ ಆರ್ autosomal recessive. ಸಿಕಲ್ ಸೆಲ್ ಅನಿಮಿಯಾ ಆಯ್ತು ಇದು. ಓಕೆನಾ? Where is that? Phenylketonuria. Very simple, autosomal recessive disease. Now in this disease what is happening is your phenylalanine is not converted to tyrosine. It is not getting converted to tyrosine. Phenylalanine amino acid is not converted to tyrosine. Why? Because the phenylalanine hydrolase enzyme is missing. If that enzyme is missing, this conversion is not happening. Now today students tell me, if it is not getting converted, this phenylalanine right here, it will look for an alternative pathway. And it will become phenylpyruvic acid. Yes. And this phenylpyruvic acid starts getting accumulated in the brain also. Nodi, accumulation of these in the brain results in the mental retardedness. Ajotege, it is also excreted through urine, that is poor absorption by kidney. It can cause so many disorders in the body because of the phenylalanine is not getting converted to tyrosine. Okay, so that was phenylketonuria. The next disease is called as thalassemia. Very simple. Thalassemia dali, I will tell you a small concept here. If you know the structure of hemoglobin, a hemoglobin has 4 chains, 2 alpha chains and 2 beta chains. A structure of hemoglobin which holds oxygen, it has 2 alpha chains and 2 beta chains. In thalassemia, what is happening is this alpha chain is missing or beta chain is missing. In thalassemia, either alpha chain ತುಂಬಾ ಕಮ್ಮಿ ಆಗೋಗುತ್ತೆ ಇಲ್ಲ ಬೀಟಾ ಚೈನ್ ತುಂಬಾ ಕಮ್ಮಿ ಆಗೋಗುತ್ತೆ. ಆಲ್ಫಾ ಚೈನ್ ಕಮ್ಮಿ ಆದ್ರೆ ಆಲ್ಫಾ ಥಲಸೀಮಿಯಾ, ಬೀಟಾ ಚೈನ್ ಕಮ್ಮಿ ಆದ್ರೆ ಬೀಟಾ ಥಲಸೀಮಿಯಾ ಅಷ್ಟೇ. ಸೇರ್ ಆಟೋಸೋಮಲ್ ಲಿಂಕ್ ರೆಸೆಸಿವ್ ಡಿಸೀಸ್ ಅದು ನಿಮ್ಗೆ ಗೊತ್ತು. ಬ್ಲಡ್ ಡಿಸೀಸ್ ಇದು ಕೂಡ ಲೈಕ್ ಜಸ್ಟ್ ಲೈಕ್ ಸಿಕಲ್ ಸೆಲ್ ಅನಿಮಿಯಾ, ಬ್ಲಡ್ ಡಿಸೀಸ್ ಇದು. The defect could be due to the either mutation or deletion of which ultimately results in the reduced rate of synthesis of one of the chains. ಹೇಳಿದ್ನಲ್ಲ ಯಾವುದೋ ಒಂದು ಚೈನ್ ಮಿಸ್ಸಿಂಗ್ ಇರುತ್ತೆ, ಆಲ್ಫಾ ಇಲ್ಲ ಬೀಟಾ ಚೈನ್. So based on that we have alpha thalassemia and beta thalassemia. This causes a formation of abnormal hemoglobin molecules resulting in anemia, which is a character of this disease. Obviously alpha chain missing or beta chain missing andre hemoglobin is not functioning properly now, not functioning properly. Okay. Now what is alpha thalassemia? Understand, alpha thalassemia is controlled by two closely linked genes, HbA1, HbA2, hemoglobin A1, A2 on chromosome 16 of each parent. So one parent hatra ಎರಡು gene ಇದೆ, so totally 4 genes. One parent hatra 2 genes, two parents hatra 4 genes. Okay, so there are 4 genes. Now what happens is due to the mutation or deletion of one or more than 4 or of the 4 genes, ಈ HbA1 HbA2 ಜೀನ್ ಅಲ್ಲಿ ಮ್ಯೂಟೇಷನ್ ಆದ್ರೆ ಏನಾಗುತ್ತೆ, ಆಲ್ಫಾ ಚೈನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲ್ಲ. The more genes affected, less alpha chain, more molecules produced, less alpha chain. So that causes alpha thalassemia. If these 2 genes, chromosome 16, HbA1 HbA2 in one parent ಬಂದ ಬರೋದು, that is for example this one 16 chromosome, 16 got from your mother, same 16 chromosome coming from your father also, no, in that also if there is a defect, gone case, alpha thalassemia. What is beta thalassemia? Beta thalassemia is controlled by a single gene now, single gene, HBB gene on chromosome 11, okay, of each parent. Akasmati mutation, akasmat ee chromosome 11 alli mutation ಆದ್ರೆ, ಈ gene alli mutation ಆದ್ರೆ beta chain produce ಆಗಲ್ಲ. That's how we get beta thalassemia. Beta thalassemia. Okay, thalassemia differs from sickle cell anemia in that former is a quantitative problem. Thalassemia is a quantity. Alpha chain produce ಆಗ್ತಾ ಇಲ್ಲ, beta chain produce ಆಗ್ತಾ ಇಲ್ಲ, that is a quantity problem. While sickle cell anemia was a quality problem. One small base pair change ಆಗಿತ್ತಲ್ಲಿ, that was a quality. This is quantity problem. Okay, quantity and quality. Now that is the end of all the different examples of Mendelian disorders. Now we come to something called as chromosomal disorders. Now before we go to chromosomal disorders, there are 2 small concepts which you need to remember. That is called as your aneuploidy and polyploidy. Now what is the difference? Aneuploidy is gain or loss of chromosomes Down syndrome, Turner syndrome. Illi aneuploidy illi one chromosome is added or one chromosome is removed, 1 or 2 maximum or 3. Okay, so monosomy, trisomy you can learn here. Okay, now what is polyploidy? Increase in the whole set of chromosome. Artha eno? For example, one organism is 2n, the organism can become 3n or 4n. For example, here we have 22 chromosomes, it will become 44 chromosomes here. One entire set of chromosomes are double ಆಗ್ತಾ ಇದೆ. That is called as polyploidy. Now under aneuploidy we have different types again. Okay, failure of segregation of chromatin during cell formation results in the loss of gain of electron, called as aneuploidy. All of you know. For example, Down syndrome results in the gain of extra chromosome of 21. ನೋಡಿ, extra chromosome add ಆಯ್ತಾ, aneuploidy. Addition of chromosome. Okay, aneuploidy. Polyploidy, failure of cytokinesis after telophase of cell division results in the whole set in increase of chromosome. Whole set increase adre, that is going to be 2 and 4 in ಆಗ್ಬಿಟ್ರೆ, that is polyploidy. Okay, now total number of chromosomes in normal human is 23. 22 autosomes and a pair of 6 chromosome, XXL XY irutte. Okay, now students, what if I add one extra chromosome? One extra chromosome is added. One example is trisomy. One extra chromosome is added. Trisomy is a type of aneuploidy. Trisomy of 21 is a type of aneuploidy. Okay, type of aneuploidy. First one is Down syndrome. ಈ ತರ ಮಕ್ಕಳು ನೋಡಿರಬಹುದು. This is called as Down syndrome. Now this was first discovered by Langdon Down It was first discovered by your Langdon Down. Okay. Down syndrome is genetic disorder presence of additional copy of chromosome. Langdon Down was the scientist who discovered it. Now, in the case of Down syndrome, there is an extra chromosome at the 21st position of chromosomes. Okay. So, of chromosomes number 21, one extra copy irutte called as trisomy of 21. One extra chromosome ಇದೆ ಇಲ್ಲಿ trisomy of 21. A one extra chromosome ಬಂದಿದ್ದಕ್ಕೆ ಏನಾಯ್ತು ಇಲ್ಲಿ? Because of one extra chromosome on a 21st position, because of the one extra chromosome, short head ಇರುತ್ತೆ ಮಕ್ಕಳಿಗೆ with small round head, furrowed tongue, partially opened mouth, palm is ತುಂಬಾ broad ಆಗಿರುತ್ತೆ and palm creases, ಕೈ ಮೇಲೆ lines ಇರುತ್ತೆ, because of one extra chromosome. Physical, psychomotor and mental development is completely retarded here. That's it, development agala. That is the Down syndrome. Next is Klinefelter syndrome. Klinefelter syndrome is again an additional copy of X chromosome. That is, usually males are XY normally, but in the case of Klinefelter syndrome, the males are XXY, one extra X chromosome baratte, additional X chromosome. Okay, because of that additional X chromosome, such individuals have overall masculine development but will have female to feminine development also. Development of breast called as gynecomastia. In such individuals are sterile also. Sterile. Now, what is Turner syndrome? In Turner syndrome, one X chromosome is missing. One X chromosome is completely missing here. For example, a normal female is XX. The female in Turner syndrome will be XO. One X chromosome is missing in the case of Turner syndrome. And because of that, the female with Turner syndrome are sterile as ovaries are rudimentary. Besides other features lack of other secondary sexual characteristics are missing here. So that is the three different types of chromosomal disorders. Down syndrome because of additional copy of 21st chromosome. Klinefelter syndrome, extra X chromosome. Turner syndrome, one X chromosome is missing, completely missing. And with that, students, that is the end of this lecture. Now, students, let me know in the comment section today itself that if you need a separate session of almost last 10-year PYQs from this chapter. If yes, tell me in the comment section if you want the last 10-year PYQs. And also quickly smash the like button and also let me know in the comment section if you have any doubt. If there is no doubt, just say principles of inheritance done and dusted. Full agoytidu. Okay. So with that, students, I will see you in the next video. Bye bye. Take care and have a great day.